Kutlu. Yıldız Teknik Üniversitesi Biyoteknoloji ve Genetik Kulübü olarak bu sene 4.'sünü düzenlediğimiz Astrobiology konferansına hoş geldiniz. Hepinizi aramızda görmekten büyük mutluluk duyuyoruz. Etkinliğimize başlamadan önce sizlere yapacağımız çekilişten bahsetmek istiyoruz. Gün sonunda Çarkıfelek çekilişi düzenleyeceğiz ve 2 şanslı katılımcımız 250 liralık Sinemaksimum Movie Pass paketi kazanma fırsatına sahip olacak. Etkinliğe katılım belgesini almaya hak kazanabilmek için oturum sonlarına sohbet kısmından paylaşılacak olan link bağlantılarından oturuma katıldığınıza dair doldurmanız gereken forma ulaşabilirsiniz. Konferansımız boyunca 9 oturumdan en az 7 oturuma katılmış olan kişiler katılım sertifikası almaya hak kazanacaktır. Çekilişe katılmak için ise canlı yayınımıza sohbet kısmından paylaşılacak olan çekiliş formunu doldurması yeterli olacaktır. Ayrıca bugünkü oturumların tümüne katılmış olan katılımcılarımız çekilişe katılmaya hak kazanacaktır. Şansınız bol olsun. Şimdi sizlere program akışını özetlemek istiyorum. Konferansımız bugün toplam 5 oturum olmak üzere saat 10'dan 16'ya kadar sürecektir. Oturumlar arası 10'ar dakika molalarımızın yanı sıra öğlen 1 saatlik aramız olacaktır.
Esteemed participants, greetings. I'm Berilak Gök from Team Origins of Life. Welcome to the Astrobiology and the Origins of Life conference, organized by the Biotechnology and Genetics Club of Yıldız Technical University. We are pleased to extend a warm welcome to each one of you. Before we begin our event, we would like to introduce our raffle. At the end of the day, we will hold a Wheel of Fortune draw, where 2 lucky participants will win a 250 Turkish liras worth of Cinemaximum Movie Pass package each. To be eligible to receive the certificate of participation in the event, you can access the form you will need to fill out for participating in the session from the links that will be shared in the chat section at the end of the sessions. People who have participated in at least 7 sessions out of the 9 sessions during our conference will be eligible to receive a certificate of participation. To participate in the draw, it will be enough to fill out the draw form, which will be shared from the chat part of our live broadcast. In addition, our participants who have participated in all of today's sessions will be eligible to participate in the draw. May you have lots of luck. I'll present an overview of today's schedule for you. The conference will continue across 5 sessions, spanning from 10 a.m. to 4 p.m. today. In addition to 10-minute breaks between the sessions, there will be a 1-hour lunch break.
Bugünün ilk konuşmacısı olan Sayın Profesör İrep Gözen'i, İkinci boyuttan üçüncü boyuta düz ülkede proto hücre dönüşümleri konu sunumunu yapması için davet ediyorum.
I now extend an invitation to our inaugural speaker, Professor Gözen, to deliver her presentation on From Second Dimension to the Third Dimension, Protocell Transformations in Flatland.
Aydınlar. Slide'ımı görebiliyor musunuz? Ben sizi görmüyorum, o yüzden evet derseniz çok sevinirim.
Şu an gözüküyor.
Aynen.
Ve duyuyorsunuz da böylelikle.
Evet.
Peki, çok teşekkür ederim davetiniz için. Günaydınlar, İskandinavya'dan selamlar. Konuşmamın başlığı ikinci boyuttan üçüncü boyuta düz ülkede proto hücre dönüşümleri. Buradaki düz ülkede aslında Edward Abbott'un Flatland kitabına bir selam gönderme olarak düşünebiliriz. O da Türkçe'ye çevrilen bir kitap ve düz ülke olarak çevrilmiş. Orada da sadece 2 boyutlu bir dünyada ki bir yaşamı, bir hikayeyi anlatıyor.
Hocam çok özür dileyerek sunumunuz sanırım gözükmüyormuş. Tekrar paylaşmanızı rica edebilir miyim?
Tabii. Hı.
Tamamdır. Şu an gözüküyor ekranınız. Teşekkürler.
Gözüküyor, tamam. Çok sevindim.
Evet.
Evet, şu an ikinci slayda geçtim. Onu da görebildiniz mi?
Evet.
Tamam,
evet.
Harika. Evet, konuşmama bu aslında bir NASA Araştırma Merkezi'nin duvarında olan bu resimle başlamak istedim. Çünkü bu resim işte dünyanın oluşumundan bugünkü hayata kadar olan bir süreci resmeden bir resim. Ve yaklaşık işte 4,5 milyar, işte 4,6 milyar yıllık bir süreci anlatıyor aslında tek bir resimde. Bu ortadaki beyaz çizgiyi de bir zaman çizelgesi olarak düşünebilirsiniz aslında. İşte sol taraftan sağ tarafa doğru ilerliyor zaman ve sol tarafta işte en arkada, hatta daha da eski Big Bang, büyük patlama, işte genç dünyanın oluşumu, volkanik aktiviteler ve bir süre sonra sulu ortamda moleküllerin varlığı, bu moleküllerin bir araya gelerek oluşturdukları yapılar, hücre formuna geçiş ve daha kompleks canlılara geçiş, sağa doğru gittikçe artık iyice hani köpek balıklarını, dinozorları, hatta ilk insan bile resmedilmiş, görebilirsiniz sağ tarafa doğru. Ben bu sürecin tam olarak şu noktasına bakıyorum, bu okla gösterilen noktaya. Bu da işte bu moleküller karışımının diyeyim bir şekilde bir kimyasal karışımdan yaşam denilebilecek bir forma adım attığı tam o kısma denk geliyor. Ve işte bildiğimiz gibi hayatın nasıl başladığını sorguluyoruz. Bunu henüz bildiğimiz ve cevaplayabildiğimiz bir soru değil bu. Ve şu an aslında bilimin cevaplayamadığı 10 büyük sorudan biri olarak düşünülüyor, biliniyor. Ve bu sorunun altında daha spesifik başka sorular var. Bunlardan bazıları mesela bu ilkel hücrelerin nasıl oluştukları ve nasıl kopyalandıkları, nasıl çoğaldıkları, içindeki genetik polimerlerin nasıl kapsüle edildiği, sonra bu yapının nasıl büyüyüp bölündüğü gibi sorular ve ben aslında bu ilk soruya biraz odaklanmış durumdayım araştırmalarımda. Yani o ilk oluşan yapının nasıl oluştuğuyla ilgili aslında benim araştırmalarımın çoğu. Şimdi kimyasal bir birimden, yani sadece kimyasal moleküllerin toplanmasından oluşan bir birimden yaşıyor denilen bir forma nasıl geçildiğini araştırmak için biraz yaşamın ne olduğunu da, yani o tanımın üzerinde de durmak gerekiyor. Ve bir sürü farklı bilim insanının yaptığı bir sürü farklı tanım var yaşamın ne olduğuna dair. Benim çok sık başvurduğum tanımlardan biri bu Tibor Gantin'in aslında 60'ların sonunda kurduğu bu kematon modeli ve bu modelin anlamı şu, yani bir formun yaşıyor denilebilmesi için sahip olması gerektiği 3 minimum özelliği aslında tanımlıyor. Bunlardan birincisi bariyer, yani o yaşam formunu yaşamayandan ayıran fiziksel bir sınır. Daha sonra bu sınırın içinde işte o sınırdan geçen molekülleri bir şekilde ayrıştıran bir metabolik birim ve enerjiye dönüştüren bir biyokimyasal reaksiyonlar zinciri daha doğrusu. Ve hücrenin bu süreç içinde edindiği bilgileri işte saklayabileceği, tekrar kullanabileceği bir bilgi işlem merkezi diyeyim tırnak içinde. Yani genetik bir depo merkezi aslında. Dolayısıyla biz hani deneyciler olarak alanda bu hücreyi, yani yaşamsal ilkel bir hücreyi ilk taklit etme aşamasında aslında o sınırı oluşturmaktan çoğumuz başlıyoruz. Bu hani bizim için en pratik adımlardan biri ve bu kompartmanı oluşturmak için bir sürü bunu farklı materyalden ve farklı protokolle yapabilirsiniz. Başka modeller var alanda, farklı araştırmacıların kullandığı. Bu işte conservate'ler olabilir, polimerik vesiküller olabilir veya çokça insanın kullandığı, bu slaytta da işte bu resmetmeye çalıştığım lipid vesikülleri olabilir. Bunların çok fazla kullanılmasının aslında en önemli nedeni şu anki modern hücrelerin de bu yağ membranıyla çevrilen aslında bir zarfın içinde olması. Yani eğer biz bugünkü hücreyi anlamaya ve onun nereden geldiğini bulmaya çalışıyorsak aslında ona çok yakın bir modelden de başlamış oluyoruz bunları kullanarak. Ve dediğim gibi hani bu alanda tam çalışmayanlar için açıklamak gerekirse bu yağ moleküllerinden oluşuyor. İşte yağ molekülleri sulu bir ortama konulduğunda kendileri organize olup bu kompartmanları, bu küresel kompartmanları, işte vesikül dediğimiz yapıları oluşturuyorlar. Bunlar tek katman olabiliyor veya birçok membranın üst üste istiflenerek oluşturduğu bu çok katmanlı vesiküller olabiliyor. En aşağıdaki panellerde bunların mikroskop altında görünen hallerini görüyorsunuz. En soldaki iki panel çok katmanlı vesiküller ve sağdaki aslında biri diğerinin renklerinin tersi çevrilmiş olanı daha kolay görünmesi için o da tek katmanlı bir vesikül. Bu arada bu slaytta şeyi de söylemekte fayda var. Bugün size aslında anlatacağım her şey, deneysel olan her şey sulu bir çözeltinin içinde geçiyor. Çünkü resimlerde tam o su kısmı yansımayabiliyor ve bazen bir karışıklığa neden olabiliyor. Ama anlattığım her şey, yani yüzeyler de dahil olmak üzere her şey sulu bir ortamda. Çünkü bizim bildiğimiz hayat suya dayalı. Şimdi bu aşamada genelde bu vesikülleri yapmaya yarayan, işte yapmak için kullandığımız daha doğrusu protokoller çok yerleşik protokoller. Yani uzun yıllardır kullanılıyorlar, verimi yüksek protokoller. Yani neredeyse çok büyük bir yanlış yapmadığınız sürece her zaman vesikül oluşturabiliyorsunuz laboratuvarda. Modifiye edebiliyorsunuz bu protokolleri, yani işte içine kullandığınız floresan boyayı değiştirebiliyorsunuz, lipidlerin kompozisyonunu değiştirebiliyorsunuz, içine kapsüle edebiliyorsunuz belli bileşenleri. Dolayısıyla burası aslında biraz işin yani alandaki araştırmacılar tarafından en sıkıcı ve basit bulunan kısmı. Aslında herkes bunu yapıp bir sonraki aşamaya bakmaya yönelik araştırmalar yapıyor ama ben burada biraz daha zaman harcamamız gerektiğini düşünüyorum. Onun nedeni de şöyle belki açıklayabilirim. İşte şu andaki alandaki yaklaşım biraz modern hücrelere bakıp, eğer modern hücreyle akıllı telefon arasında bir analoji kurarsak, herkes işte o modern hücreye bakıp ya işte bunun bunun ilkel formunun her ne ise onun da muhtemelen bir ekranı var, onun da bir kamerası var, içinde uygulamalar var falan gibi bir düşünceyle geliyoruz aslında. Ama biraz akıllı telefonlardan zaman olarak geriye gittiğimizde işte tuşlu cep telefonları vardı. Ondan önce işte sabit telefonlar vardı, kordonlu, tuşlu veya çevirmeli. Oradan daha da en eski, artık telefonun en eski bu Graham Bell'in icat ettiği bu mikrofon ve telefonik forma baktığımız zaman aslında bu akıllı telefonlarla arasında hiçbir benzerliği olmayan bir formla karşılaşıyoruz. Biraz benim araştırmalarımın üstünde, yani baktığı perspektifte böyle. Yani bugün biz modern hücrelere bakarak hep modellemelerimizi deneysel olarak öyle yapıyoruz. Ama böyle yapmasak aslında hani 4 veya 3,5 milyar yıl önceki o proto hücreler bambaşka morfolojilerde olabilir mi? Ve biliyoruz ki biyolojide genelde yapı fonksiyonla eşleşen bir kavram. Yani bir şeyin işte yapısının belli bir formda olması onun aslında belli bir fonksiyonu da yapabilmesini mümkün kılıyor. Dolayısıyla aslında cevaplamaya çalıştığım soru bu en eski hücre formları hangi morfolojilerdeydi ve bu onların çeşitli çevresel etkiler altında neleri başarmasını sağlayabilirdi gibi bir yerden bakıyorum aslında araştırmalara. Ve şimdi biraz önce size o solüsyonlar içinde, yani suya koyduğunuzda organize olan yağ moleküllerini anlattım. Bunları bir de yine sulu ortamda ama bir katı yüzeye koyduğunuzda ne olduğunu göstereceğim. Şimdi aşağıdaki videoda bu genişleyen bir yuvarlak bir yapı görüyorsunuz. Bu mikroskop altında aslında bu çok katmanlı vesikülün davranışı. Ve ortada o gördüğünüz parlak yapı, yani o en dairenin ortasındaki o parlaklık aslında çok katmanlı vesikül, yani içinde bir sürü membran olan bir lipid deposu gibi düşünebilirsiniz bunu. Ve yayılan o daire de aslında iki membranın üst üste gelmiş şekli. Ve bu genişliyor ve üstteki animasyonda da o genişleyen ucun resmedilişini görüyorsunuz. Yani o kenarlar, o dairenin uçları bu şekilde, yani aslında tank paleti hareketi gibi bir şey deniyor alanda buna. Bu yayılmayı sağlayan, yani buna katkıda bulunan daha doğrusu bir sürü parametre var. İşte membranınızın elektro yükü, yüzeyinizin yükü, ortamda kalsiyum, magnezyum gibi iyonların olup olmadığı, ama en etkili, yani bu hareketi en domine eden faktörlerden biri yüzey gerilimi. Onu da en basit şekilde şöyle anlatabilirim. Şimdi sizin vesikülünüzün katı yüzeyle fiziksel temas kurduğu anda aslında orada su var. Ve suyun yüzey gerilimi yağdan daha yüksek, daha kuvvetli. O yüzden aslında yağ yüzeyle temas haline geçtiği anda su kendini topluyor ve onun topladığı yere bu yağ yayılıyor. Yani aslında tamamen yağ ve suyun katı ara yüzeydeki yüzey gerilimlerinin farklı olması bu yayılmayı sağlayan hareket. Bu yayılmanın sonucunda bir sürü farklı morfolojiye doğru gidebiliyorsunuz. Yüzeyinizin veya lipidlerinizi değişimi sayesinde. Bu slaytta gördüğünüz bütün yapılar benim araştırmalarımda gözlemlediğimiz yapılar. Ve bunların iki ortak özelliği var. Biri işte yağ moleküllerinden yapılmış olmaları ve kendi kendilerini oluşturmaları. İki, bunların hepsinin katı yüzeylerde iki boyutta oluşup oradan üçüncü boyuta gelişmeleri, evrilmeleri daha doğrusu. Peki yüzeyler buna nasıl sebep oluyor dersek, onu genelde şöyle bir örnekle açıklıyorum ben. Katı bir madde bloğunu, hatta bu yani tebeşir gibi bile düşünebilirsiniz. Bunu ikiye kırdığınızda aslında iki yeni yüzey oluşturuyorsunuz. Ve o oluşturduğunuz yüzeydeki atomlar maddenin içindeki atomlara göre şöyle bir farkı oluyor dolayısıyla. Maddenin içindeki atomlar kendi gibi atomlarla çevriliyken ve onlarla bağ kurmuşken kırdığınız yüzeyde artık bir tarafta bu atomlardan yoksun kalmış oluyor. O tam yüzeyde oluşturan ya da kalan atomlar. Ve bu yüzeyleri oluşturmak için siz aslında iş yapıyorsunuz ve birim yüzey başına yaptığınız iş aslında bu yüzey gerilimini, daha doğrusu yüzey enerjisini tanımlıyor. Ve sistemler her zaman enerjilerini minimize, yani yüzey serbest enerjilerini minimize etmek üzerine bir eğilimleri var. Bu şu anlama geliyor. Yani yüzeyler oluşturdukları anda aslında bağışlamak istedikleri bir enerjiye sahip oluyorlar. Bu neden önemli hayatın başlangıcı probleminde? Şu yüzden önemli. Çünkü eğer siz daha kompleks biyolojik yapıları oluşturmamışsanız, ilkel bir hücre yapısındaysanız, işte proteinleriniz, ATP, gerekli sofistike metabolik reaksiyonlarınız daha yerleşmemişse, yüzeylerin bize sağladığı enerji aslında bu yağ moleküllerinin oluştuğu hücre modellerine ve onların dönüşümlerini gerçekleştirebilecek enerjiyi sağlayabiliyor yüzeyler. Aslında bu yüzden önemli. Ve yüzeyler dünyanın oluşumunda aslında ilk oluşan şeyler. Yani su ve biyolojik materyaller onlardan çok sonra geliyor aslında. Dolayısıyla yüzeyler çokça varlar ve çoğu da yüksek enerjili yüzeyler ve bu aslında hayatın başlangıcı probleminde işte adsorpsiyon, kataliz, yani prebiyotik reaksiyonların katalizi falan bağlamında düşünülmüş faktörler, bileşenler, ama bu proto hücre bağlamında yüzeylerin etkisi pek önemsenmemiş veya düşünülmemiş. İşte ben de biraz o konuya eğilmiş bulunuyorum. Şimdi size deneysel olarak bu yüzeylerin neler yaptırabildiğini proto hücreleri anlatacağım, ama ondan önce hangi yüzeyleri kullandığımızı biraz açıklamak isterim. Birincisi sentetik yüzeyler. İşte bu sağ üst panelde de gördüğünüz, resmini gördüğünüz temiz oda denilen laboratuvarlarda bizim oluşturduğumuz yüzeyler. Bu temiz odalarda da çalışmayanlar için çok basitçe açıklamam gerekirse işte içindeki hava filtre edilen, yoğun bir şekilde filtre edilen odalar. Mesela çok yoğun bir şehir ortamında 1 metreküpte sizin 30 milyon partikülünüz varsa Bu temiz odalarda bu sayı 30 gibi bir sayı. Dolayısıyla işte adı da temiz oda adı oradan geliyor zaten. Ve bu tabii atomik olarak bir kesinlikle ve saflıkta bir yüzey oluşturmanıza yardımcı olan ortamlar. Bu tabii deneysel bağlamda önemli çünkü yani tamamen özelliklerini bildiğiniz, karakterizasyonu full olarak yapabildiğiniz, bütün olarak yapabildiğiniz yüzeylerle çalışıyorsunuz. Oradan elde ettiğimiz sonuçları da daha sonra doğal yüzeylere aktardık. Doğal yüzeyler derken de bunlar mineraller, kayalar, kayaların içinde de birçok mineral var veya camlar. Ve bunlar hayatın başlangıcıyla aslında ilişkili, yakın ilişkili hatta yüzeyler. Çünkü hayatın başlangıcında oluşan birçok yüzey aslında şu an dünyamızda da şu an yaşadığımız çevrede de varlar yani. Ve en son araştırmaların artık geldiği noktada benim laboratuvarımda uzaydan gelen yüzeyleri kullanmak. Çünkü uzaydan da dünyamıza bir sürü göktaşı geliyor. Ve onlar da aslında katı yüzeyler. Ve o yüzden onları da kullanıyoruz. Ve burada şöyle de bir düşünce var. Bu göktaşları sadece uzaydan bizim dünyamıza gelmiyor. Başka gezegenlere de gidiyorlar. Ve başka gezegenlerde de lipid molekülleri var. Çünkü bunu da göktaşlarının içindeki analizlerden biliyoruz. Yani göktaşların içindeki organik moleküllerin repertuarı çıkarıldığında burada lipidler var ya da lipid benzeri basit moleküller var. Dolayısıyla aslında bizim dünyamıza gelen göktaşları ve lipidler başka gezegenlere de ulaşıyorlar. Dolayısıyla bizim burada laboratuvarda elde ettiğimiz sonuçlar eğer başka gezegenlerde de su ve işte bizim dünyamıza benzeri şartlar varsa orada da oluşabilecek yaşam formlarını bir anlamda anlamış oluyoruz. Şimdi deneysel bulgular üzerinden devam edecek olursak size yayılmayı göstermiştim yüzey üzerinde. Burada şunu anlatacağım ki o yayılma sonsuza kadar devam etmiyor. Çünkü o sizin o katmanlar, o iki katman, yayılan katman aslında ortadaki çok katmanlı bu vesikülden çıkıyorlar. Yani o lipidler ve o vesikülde bir hani sonuçta sonlu bir yapı ve içindeki katmanlar tükenebiliyor veya bu yapı çok genişlediğinde o vesikülden o artık yapının uçlarına lipid o kadar kolay akamamaya, o kadar hızlı akamamaya başlıyor. Dolayısıyla o tansiyon çok artıyor ve bir şekilde o kırılma tansiyonunun üstüne çıktığında da bir yırtılma görüyoruz. Şimdi burada onun videosunu göstereceğim size. Yine mikroskop altında üstten bakıyorsunuz gibi düşünün. Bu işte çiçek şeklinde bir yırtılma oluyor. Yani bu bir delik aslında ve siz bu delikten yukarıdaki katman açılırken aşağıdaki katmanı görüyorsunuz. Bunun renkli olmasının nedeni işte bu aslında kırmızı değil, bu yani yeşil, sarı bir sürü renk buna atayabilirsiniz. Ama floresan bir boyayla bu lipidleri biz boyadığımız için bunu floresan bir mikroskopun altında görebiliyoruz. Ve işte daha kırmızı olan kısım iki katmanlı olan yer, daha bordo gibi daha koyu renk olan kısımda aşağıdaki yani yüzeyin tam üstündeki membran, çevresindeki siyah kısımda çıplak yüzey. Yani buna bakıyorsunuz yukarıdan gibi düşünün. Benim işte doktora sürecimde ben bu yırtılmaları karakterize etmeye çalışıyordum. Çünkü o zaman bilinmiyordu hücre zarlarının bu yırtılmaları yapabileceği. Dolayısıyla ben sadece bunlara yoğunlaşmıştım. Ve genelde bu yırtılma olduğu zaman yani şöyle videodan beni görebiliyorsunuz eğer aslında şöyle iki katman var ve ortadaki açılıp böyle yüzey üzerine şöyle yayılıyor. Yani bir böreğin üstündeki yufkanın açılıp şöyle tezgahın üzerine açılması gibi düşünebilirsiniz belki bunu. Ve ben bu noktadan sonra hani yırtılma artık devam etmediği için çünkü yüzeyin üstündeki membran hiçbir zaman yırtılmıyor. O yüzeye çok kuvvetlice yapıştığı için sadece onun üstündeki ikinci katman yırtılıyor kenarlardan çekildiği için. Dolayısıyla ben hani bu noktada artık bu deneyin devamına pek bakmıyordum ama benim ilk öğrencim İlayda Kantarcı bunu gece yani deneyini yaptıktan sonra bırakıyor ve sabah baktığında böyle tek katmanlı vesiküller görüyor. Ve bunları biz karakterize etmeye başladık. İşte tam bu zaman noktasında baktığınızda yaklaşık bu bir yayılmadan çıkan 1000'in üzerinde vesikül oluştuğunu görüyoruz. Ve bunların çapları işte 5 mikrometre civarında. Yani gayet biyolojik hücrelerle karşılaştırılabilir bir boyutta. Ve bunları birkaç saat bıraktığınızda eğer çevrede multivesiküler yapılara ulaşabilirliği varsa bu yapının daha da büyüyorlar. Yani ve böyle sınırları birbirine değecek kadar bu yuvarlak şeyi dolduruyorlar, sınırı dolduruyorlar. Burada başka bir yani confocal microscopy resmi bu. Yani sanırım Türkçe'de eş odaklı mikroskop deniyormuş. Tam emin değilim. Bu arada araştırmalarımı ilk defa Türkçe sunuyorum. O yüzden bazı terminolojide bazı hatalarım olursa kusura bakmayın. Ama kontrol ettiğimde yani öyle bir karşılık buldum. Bu eş odaklı mikroskopun özelliği şu: siz 3 boyutlu bir numunede bir sürü kesit alabiliyorsunuz yüzeyden yukarı doğru ve o kesitleri böyle istifleyerek 3 boyutlu bir resim çıkarabiliyorsunuz ortaya. Burada gördüğünüzde o resimlerden biri. Şimdi burada ne görüyoruz? Sol üst köşede, bilmiyorum mouse'umun cursor'ını görebiliyor musunuz ama burada biraz daha açık renkli bir bölge var. Bu beyaza yakın bir bölge. Orası hala da 2 katmanın üst üste olduğu bir bölge. Bu daha koyu gri füme alttaki resim, alttaki membranın floresanı aslında. Ve onun üstünde böyle ipliksi ağ gibi yapılar görüyorsunuz. Bunlar nano boyuttaki tüpler. Bunları daha çok açıklayacağım ileriki slaytlarda. Ve bunun üstünde de işte tek katmanlı bu vesikülleri görüyorsunuz. Yüzeye yapışmış bir şekilde duran. Böyle bir sürü mikroskop deneyi ve bir sürü resmi inceledikten sonra farkına vardık ki bu vesiküller aslında bu küçük iplikçiklerden tomurcuklanarak büyüyorlar. Burada mesela çok küçük olanlarını, daha yeni oluşmuş olanlarını bu slaytta görebilirsiniz. Ortasında biraz daha yuvarlak bir yapı, kenarlarında daha sivrilen ve o tüplere bağlanan kısmı görüyorsunuz. Ve bu oluşumu aslında video olarak yakalamayı başardık. Yani yırtılma, tüplerin oluşması ve o tüplerden bu tomurcukların çıkması gibi. Onu da bir sonraki slaytta burada gösteriyorum. Yine yukarıdan bir membranın, yani yayılmış bir bu hücre zarlarının üst üste olduğu bir bölgeye bakıyorsunuz şu anda. Ve üstteki yırtılmada işte üstteki membranın çekildiğini, alttaki membranın üzerinde çekildiğini görüyorsunuz. Çekilirken bu tüpler aslında spontane olarak oluşuyorlar. Ve aslında bu kesitin alt sol kısmına baktığınızda küçük tomurcuklanmaları görebilirsiniz tüplerden gelen. Şimdi bu yapıyı birkaç saat bıraktığınızda çevredeki o sulu çözeltiyi içine alarak işte swelling dediğimiz İngilizce'de ya da şişerek diyeyim artık daha büyük işte 5-10 mikrometre hatta daha büyük boyutlarda vesiküllere doğru büyüyorlar. Peki bu tüpler neden oluşuyor derseniz bunların yani birçok oluşma pathway'i, işte yolculuğu olabilir, yolu olabilir ama biz bir tane önerdik. Bunda da şöyle düşünüyoruz. Aslında bu 2 katmanın birbirine geri dönüşümsüz olarak yapıştığı yerler var. Bu da en azından bizim deneysel sistemimizde kalsiyumlu oluyor. Şimdi kalsiyumun hem teorik, yani bilgisayar simülasyonlarıyla anlaşıldığı haliyle hem deneysel olarak bulguların gösterdiği kalsiyumun böyle bir etkisi olduğu biliniyor zaten. Yani lipid baş gruplarına bağlanıp aynı anda birden çoğuna bağlanıp 2 membranı birbirine neredeyse geri dönüşümsüz olarak yapıştırabiliyor. Dolayısıyla siz üstte bir yırtılma olduğunda, o gerilim olup da yırtılma olduğunda bazı kısımlar arkada kalıyor. Böyle iplik gibi o membran aslında çekiliyor. Bu arada enerji bağlamında baktığınızda bir membranın yırtılması, kopması, yani o tübün, ipliğin uzamasından ziyade o ipliğin kopması çok daha büyük bir enerji bariyerini gerektirdiği için lipid membranları genelde deforme olmaya daha yatkınlar. Yani çekiyorlar, uzuyorlar, yapı değiştiriyorlar ama kopmak en son çareleri. Yani basitçe böyle anlatmış olayım. Dolayısıyla bu sağdaki 3 küçük panele baktığınızda aslında ilk başta o çekilmede muhtemelen düz membran yapıları çekiliyor. Ama bu enerji bağlamında çok tercih edilebilen bir şey değil. Çünkü bizim işte biyomekanik olarak membranlara baktığımızda bir kenar enerjisi denilen bir şey var. Yani bu sisteme mal olan bir enerji olarak düşünebilirsiniz bunu. Ders kitaplarında, moleküler biyoloji kitaplarında genelde bu membranların ucu bu şekilde açık olarak resmediliyor. Hani membranın yapısını anlatabilmek açısından. Ama tabii gerçekte hiçbir zaman böyle açık değiller. Çünkü sudan sakınan kısımlarını kapatmak için aslında bu üst panelde şu kutucuğun içindeki çizimdeki gibi lipid molekülleri böyle o kenarı yuvarlıyorlar. Ama bu da çok tercih edilen bir şey değil. Çünkü lipid moleküllerinin arası açılıyor ve suya daha maruz kalıyor fiziksel olarak. Dolayısıyla bu bir kenar enerjisi oluşturuyor ve bu kenar enerjisi kenarın uzunluğuyla orantılı. Yani kenarınız ne kadar açık ve uzun olursa veya kapalı yani ne kadar kıvrık ve uzun olursa o kadar sistemin enerjisi yükseliyor. Dolayısıyla bu enerjinin cezasını ödememenin tek yolu membran için aslında o kenarı kapatmak. Yani o ucu kapatmak. Dolayısıyla bunu yapabildiği formda bir tübe kapanmak olarak düşünebiliriz. Sonuçta da bu işte ipliksi böyle ağ gibi gördüğünüz bu nano tüp yapılar oluşuyor. Peki bunlar daha sonra eğer bu enerji bağlamında tercih edilen bir membran formuysa bunlar neden vesiküllere dönüşüyorlar derseniz, çünkü bu sadece aslında relatif olarak enerji açısından tercih edilebilen bir şey. Yani bu sağdaki 3 paneli düşünürsek aslında en üsttekine göre enerjisel olarak daha tercih edilen bir form. Ama yine de oradaki kıvrımın boyutu nano. Ve bu da bir kıvrım enerjisi veya bükülme enerjisi deniyor sanırım. Fizik çalışan arkadaşlar daha iyi bileceklerdir. Ve bu bükülme enerjisini bu sefer çok yükseltmeye başlıyor. Yani membranlar aslında çok büyük kıvrımlara da sahip olmak istemiyorlar. Dolayısıyla eğer lipid materyaline ulaşabilirlikleri varsa büyük vesiküllere büyümeyi tercih ediyorlar. Bunu da bir matematiksel modellemeyle aslında doğruladık. Biz genelde membranların biyomekaniğine baktığımızda bu Helfrich membran elastisite teorisini kullanıyoruz. Bu aslında fizikçiler için biraz basit ve o yüzden biraz da sıkıcı bir teori. Ama bizim çok işimize yarıyor. Özellikle bu basit hücre formlarının deformasyonlarına baktığımızda. Şimdi burada yaptığımız şey basitçe yine size anlatmam gerekirse bir nano tüp aldık ve bu tübe kademeli olarak artan miktarlarda membran materyali ekledik. Ve her seferinde de dedik ki simülasyona bunun yüzey enerjisi olarak en düşük olduğu geometrik formu bize bul. Dolayısıyla ekledikçe aslında burada rakamıyla 5 ile gördüğünüz bu forma bürünüyor. Yani deneyde aslında gördüğümüz yapılara çok benzer yapılara bürünüyor. Dolayısıyla buradan çıkaracağımız sonuç şu ki yani kıvrılma enerjisi aslında burada bu hareketi domine eden faktör. Yani önce sizin membranınız, özetlemem gerekirse siz bir çok katmanlı bir vesikülü yüzeyle temas haline geçtiğinde yayılıyor. Üst katman bir süre sonra gerilime dayanamayıp yırtılıyor ve bu iplik şeklinde olan ağlara dönüşüyor ve bu ağlardan da o kıvrımı minimize etmek adına bulabildiği yerlerden de tomurcuklanarak bu büyük vesikül yapılarını oluşturuyor. Bunu da bir kıvrım olarak düşünüyor çoğu insan ama aslında mikron düzeydeki bu vesikül yapılar neredeyse düz membranla aynı enerjiye sahip. Yani aslında o gördüğünüz kıvrım nano kıvrıma göre çok daha düşük enerjili bir kıvrım. O yüzden de sistem oraya doğru sürekli gidiyor. Bugün maalesef zamanım limitli, o yüzden size o kısmı gösteremiyorum ama mikro akışkan çipler kullanarak böyle çok basit bir akım uyguladığınızda, hidrodinamik bir basınç uyguladığınızda bu vesiküllere bunların yüzeyden ayrıldığını da ve başka yüzeylere göç ederek oralara da yapıştığını da görebiliyorsunuz. Bu da işte hayatın başlangıcı ve proto hücre anlamında tercih edilen bir şey aslında. Sadece orada mı kalıyor yoksa başka ortamlara da girebilecek yapıda mı? Şimdi buraya kadar anlattığım bulgularda tüpleri görüyoruz ve nasıl oluştuğuna dair bir teoremimiz var, bir savımız var ama tüplerin içindeki yapıları tam bilmiyorduk. Yani bu böyle boş bir silindir, içinde hani su olan, sulu bir merkezin olduğu bir silindir mi, yoksa daha misal gibi bir yapı mı? Yani tamamen içinde yağların olduğu ve kapalı bir yapı mı? Bunu anlamak için şöyle bir çalışma gerçekleştirdik. Bu vesikül tüp ağlarını hiç floresan boya kullanmadan yaptık. Yani oluşturduk önce. İşte buna da bu differential interference contrast, yani confocal mikroskopinin, eş odaklı mikroskobun bir add-on'uyla, ek bir fonksiyonuyla buna bakabiliyorsunuz. İşte soldaki panelde kesitini görüyorsunuz. Tam vesicle'ların böyle ortasından bir kesit aldığınızı düşünün, ekvatordan. O kesiti görüyorsunuz. İkinci panelde yüzeye en yakın yerden aldığımız bir kesiti görüyorsunuz. Dolayısıyla artık tüpleri ve o vesicle'ların bazını, yani yere yakın kısmını, tabanını görebiliyorsunuz. En sağdaki küçük panelde zaten ikinci paneldeki o kesikli çizgide çerçeve içine alınan yerin büyütülmüş hali. Yani tüpleri görebiliyorsunuz aslında. Ve buradan sonra şöyle bir deney yaptık. Ya floresan bir boya veya floresan boyayla boyanmış bir RNA veya DNA molekülünü bu yapıların üstüne koyduk ve bu yapılar bunu kapsüle edebiliyorlar. Özellikle bu işte mikro akışkan çipler kullanarak çevresinde bu akımı yarattığınızda bir kısmını kapsüle ediyorlar. Burada sağ üstteki 3 panelde bu enkapsüle edilmiş sanırım RNA'ydı bu örnekteki floresanlı molekül. Bunları enkapsüle ettiğini görüyorsunuz. Şimdi burada da şöyle bir deney yaptık. Buna işte fluorescence recovery after photobleaching deniyor. Şimdi Türkçesini tam şu an bilemiyorum ama şöyle anlatabilirim prensibini. Bu floresan kapsüle edildikten sonra yoğun bir lazer ışınıyla bir bölgeye, bir veya birkaç vesiküle bu ışını tuttuğunuzda içindeki floresan molekülleri geri dönüşümsüz olarak söndürebiliyorsunuz. Ve burada da şöyle bir sav kurduk. Yani bu vesiküller eğer birbiriyle bağlantılı değilse, bu kanallar kapalıysa o zaman bu sönen floresan hiçbir zaman geri kazanılmayacak. Çünkü veya işte çevreden bir sızıntı, akıntı falan varsa saatler alıyor bu. Yani o kadar hızlı bir zamanda olmuyor. Ama eğer tüpler, yani vesiküller birbirine bağlıysa ve tüpler açıksa moleküler difüzyonla hala daha floresanı aktif olan vesiküllerden, kompartmanlardan bu floresanı sönmüş olan kompartmanlara doğru bir akış gerçekleşmesi gerekiyor. Ve aynen de öyle oldu aslında. İşte bu panelde de onu görüyorsunuz. Floresanın lazerle söndürüldüğü panel J paneli ve sonra yaklaşık 2 dakika sonra içindeki floresanın tekrar geri oluşmaya başladığını görüyorsunuz. Aşağıda da tabii bu sadece bir örnek ama biz bunu 5-6 kere, sanırım 6 veya 7 grafik olması lazım aşağıdakilerin ve üstteki panellere denk gelen bu grafiklerin içinde E ile işaretlenmiş olanı. Yani yaklaşık 2 dakika sonra o yeniden geri kazanımı görüyorsunuz. Dolayısıyla bu tüplerin de aslında açık olduğunu göstermiş olduk. Ve bu tekrar işte bazı problemlerin yanıtlanmasına nasıl yardımcı olabilir diye düşünürseniz böyle bir deneysel sistem mesela şöyle düşünebiliriz. Bugün işte alanda bir bölünme konusunda bir problem var işte. Çünkü şu andaki hipotez şu ki işte bu vesiküller sulu bir çözeltinin içinde böyle suspended halde, asılı halde duruyorlar ve bir şekilde ortadan ikiye çekilip ayrılıyorlar. Ama tabii bu enerji gerektiren bir şey ve o enerjinin nereden geldiği, hani o çok kolay açıklanamayan bir şey. Var böyle deneysel çalışmalar bunu gösteren ama çok fazla varsayımlar yapmak gerekiyor. İşte pH pH birden değişiyor veya işte bir UV ışınlarının gelmesi gerekiyor. İçindeki bir kimyasalı işte bir şey stimüle ediyor falan gibi ama aslında öyle olmak zorunda da değil. Çünkü bu sistemi düşünürseniz, mesela bu A sistemini düşünürseniz, eğer vesiküllerden biri bir genetik polimeri alıp çoğalttığında bu kanallar vasıtasıyla, bu tüpler vasıtasıyla yanı başındaki başka vesiküllere bunu aktarabiliyor. Dolayısıyla siz aslında 2 tane yapısı aynı olan ve içeriği de aynı olan proto hücreniz olabiliyor. Dolayısıyla bu hani bir replikasyon, kopyalama gibi de düşünebilirsiniz. Dolayısıyla sizin gördüğünüz bu morfolojiler aslında bazı soruları farklı düşünmenize, farklı sormanıza da sebep oluyor denebilir. Ya da yardımcı oluyor denebilir. Şimdi buraya kadar kullandığımız yüzeyler bu sentetik yüzeylerdi. Tabii bu sonuçları elde edince biz doğal yüzeylere de bakmak istedik ve işte Oslo Doğa Tarihi Müzesi ve Kopenhag, Danimarka'daki Doğa Tarihi Müzesi ile ortak bir çalışma yürüttük. Buradaki işte jeoloji alanında çalışan, jeoloji alanında çalışan bilim insanlarıyla şöyle bir çalışma gerçekleştirdik. Bu yaklaşık 20 üzerinde minerali, kayayı 100 mikrometre kalınlığında böyle lam gibi, mikroskop lamı gibi ince kesitler şeklinde kestirdik mikroskop ile uyumlu olması açısından. Ve aslında aynı deneyi yaptık. Yani yüzeyi değiştirdik. Sonra bunları solüsyonlar içine koyduk. Yani tampon çözeltiydi yanlış hatırlamıyorsam okul yıllarımdan. Tampon çözeltiler içinde, yani sulu bir ortamda lipidleri de ekledik ve ne yaptıklarına baktık aslında otonom olarak. Minerallerin yanında bir de Mars'tan gelen bir göktaşına baktık ki bu çok özel bir göktaşı. Bu arada ben daha önce bu hayatın başlangıcı alanında çalışmadım. Aslında deneysel olarak gördüğümüz bulgular bizi o alana doğru itti aslında. Ve dolayısıyla ben bilmiyordum, yani bizim dünyamızda 400'ün üzerinde Mars'tan gelen aslında göktaşı varmış. Bunlar gayet kayıt altında kompozisyonları bilinen göktaşları. Bunun çok özel olmasının nedeni bu işte kendi Wikipedia sayfası olan bir meteor. Ve bu işte 400 küsür, atıyorum işte 405 olsun, 405'i bir kategori altındayken bu kendi kategorisini yaratan bir göktaşı. Özelliği de Mars'ın ilk oluşum safhasında oluşan bir yüzeyden geliyor olması. Bunu işte çeşitli analizlerle içindeki zirkon elementlerine falan bakarak anlayabiliyorlar. Ben işin o kısmında değilim, onun uzmanı değilim ama bu çok hani anlaşılabilen, artık böyle yerleşik bir analizi yapılmış bir şey yani. Şöyle de enteresan bir hikaye var. Biz işte bu meteorlar keşfedildiklerinde bir kayıt sistemi var dünyada. Ve burada mesela Wikipedia sayfasında bahsedilen Northwest Africa 7034 denilen meteor. Ama bizimkinin kodu farklı. O da Northwest Africa, şu an tam hatırlamıyorum ama işte atıyorum şu an 7544 gibi bir şey. Ve bunun olmasının nedeni de dünya, yani atmosfere girdikten sonra göktaşı ikiye ayrılıyor ve Sahara çölünde farklı noktaları, biraz uzak, yani birkaç kilometre uzak sanırım, iki noktaya düşüyor. Ve bu Wikipedia'da bahsedilen 7034 olanı, ilk önce bulunanı, bizim deneylerde kullandığımız biraz daha sonra bulunanı ve işte analizler bunların tamamen aslında match ettiğini gösteriyor ve o kırığı falan da buluyorlar. Yani bu aslında aynı göktaşından geliyormuş. Bu da ilginç bir detay. Sonuçlara dönecek olursam, bu sentetik yüzeylerde gördüğümüz birçok davranışı aslında mineraller üzerinde de gördük. Burada 4 örneğini gösteriyorum. İşte olivin, fluorite, dolerite ve pikrit, bazalt denilen 4 şey üzerinde bu yırtılmaları, ipliksi bu nanotüp ağ oluşumunu, vesiküllerin oluşumunu görüyoruz. Sentetik yüzeylerde görmediğimiz ama doğal yüzeylerde gördüğümüz ilginç bir bulgu. Bu doğal yüzeylerin çatlaklarından çıkan, neredeyse fışkıran bu vesiküller ve bu doğal yüzeylerin çatlakları yani kendilerine özgü bir şey. Bu kristal oluşma aşamasında olan bir şeymiş. Yani kristal, mesela bir mineral oluşurken birkaç noktada o kristallenme başlayabiliyormuş. Ve sonra bu kristaller birbirine doğru büyüdüğünde artık bir yerde bir fiziksel bariyer oluyor. Çünkü birbirlerine çarpıyorlar, yani birbirleriyle kontak haline geliyorlar ve orada duruyorlar. Ve dolayısıyla o çatlaklardaki yüzey enerjisi yüzeyin geri kalanına göre çok daha yüksekmiş. Çünkü atomların dizilimi orada çok daha ideal olmayan şekilde olduğu için diğer yüzeyin geri kalan kısımlarına göre. Dolayısıyla yüzey enerjisinin yüksek olduğu yerler bu çatlaklar. Oradan da bir şekilde bu vesiküllerin çok fazla oluştuğunu görüyoruz. Bunu tabii dinamik olarak, yani adım adım mikroskopla yakalamak çok zor. Çünkü oralara objektifler girmiyor ve bir 3 boyutta bir şey gözlemlemeniz gerekiyor. Dolayısıyla onu hala yakalamış değiliz, bu tam olarak bu oluşumu, ama gözlemlediğimiz bir şey bu alanlarda çok fazla tüplerin, yani nanotüplerin oluştuğu ve bunun bu işte İngilizce'de purling dediğimiz instabilitelere dönüşmesi. Yani tam fizikte ne deniyor Türkçe'de bilmiyorum ama fizikte çok bilinen bir oluşum bu. İşte bu, yani boncuk veya inci taneleri gibi böyle bir kolye gibi yapıların oluşması. Bu aslında şeyden, musluktan böyle çok ince bir su aktığında onun da böyle damlacıklara bölündüğünü görüyorsunuz. Çünkü aslında yüzey alanının hacme oranının çok yüksek olması halinde görülen bir şey. Yani sizin çok fazla uzun bir membranınız var ama içindeki hacminiz o kadar değil. O yüzden onu dengelemek adına işte böyle bir instabilite oluyor. Bu yani membranlarda da çok iyi bilinen ve karakterize edilmiş olan bir gözlem aslında. Dolayısıyla bizim sahamız da bu, hani o boncuk şeklinde gördüğünüz birbirine bağlı vesiküllerin zaman içinde yine şişerek büyüdükleri. Mars göktaşına dönecek olursak, orada da görüyoruz bu üstteki sarı çerçeve, ki o aşağıdaki GH panellerinde şu sarı çerçeveyi temsil ediyor. Gördüğünüz şey işte bu daha parlak yeşil olan kısım 2 katmanın üst üste olduğu yer. Daha koyu yeşil olan kısım sadece o yırtıldıktan sonra yüzeyin üstündeki membran. Ve burada vesikülleri ve işte tüp oluşumunu yine görüyorsunuz. Bu vesiküllerin üstünden böyle bir çizgi çektiğinizde oradaki floresan katmanına baktığınızda genelde böyle tek katmanlı vesiküllerin böyle M şeklinde bir karakterize bir tipik bir eğrisini görürsünüz. Bu pilotta da gördüğünüz gibi, üstteki grafikte yani gördüğünüz gibi. O da işte bir şöyle bir şey, küresel bir şeyin üzerinde bir kesiti aldığınızda 2 tane aslında membran geçmiş oluyorsunuz. O oralarda o intensite yükseliyor ve o yüzden böyle M şeklinde bir grafik görüyorsunuz genelde. Onu da burada görüyoruz. Peki Mars göktaşının içindeki elementlerin bir özelliği var mı? Yani belli yerlerde toplanıyor mu bu yırtılmalar, bu membran yayılmaları diye baktık. Buna da işte X ışınları ve elektron mikroskobunun bir kombine edilmiş bir yöntemle buna bakabiliyorsunuz. Yani yüzeyin elemental haritasını aslında çıkarabiliyorsunuz. Çünkü biliyorsunuz hani Mars'a kızıl gezegen de deniyor. Çünkü hani demirin yoğun olduğu bu hematit mineralleri var. Buna da baktığımızda yani baktık ve işte sol altta G panelinde gördüğünüz demir, alüminyum, kalsiyum ve magnezyum çeşitli renk kodlarıyla gösteriliyor. Ama hani bu alanlarda özel bir oluşum veya bu alanlardan sakınan bir oluşum görmedik. Genelde silikon ve oksijenin olduğu her yerde membran yayılıyor. Bunu gördük. Çok kısa biyo çeşitlilikten bahsetmek istiyorum. Çünkü bir yaşam formunun, yani bir yaşam formu oluşmazdan önce aslında çeşitliliğe çok önem vermiyoruz biz. Yani ancak yaşam ve hücreler oluşacak ki bunların çeşitliliği olacak. İşte oradan da çeşitli bu selection pressures dediğimiz, yani seçim baskıları, yani çeşitli çevresel faktörlerin değişmesine ve bunun işte hücrelerin ve o çeşitliliğin buna nasıl cevap verdiği, işte doğal seleksiyon falan gibi şeyleri o zaman tartışıyoruz. Ama bu eğer ilkel hücrelerde bu çeşitlilik acaba nasıl sağlanabiliyor diye bakarsak çok fazla bunu yani bunun uygulanabildiği deneysel sistemler yok. Çünkü siz eğer çözelti içindeki 2 proto hücreyi alıp bunları birbirine çarpıştırırsanız pek bir şey olmuyor. Çünkü bunların birbirleriyle yani etkileşimini oluşturacak enerji bariyerleri çok yüksek aslında. Ama yüzeyler üzerinde öyle olmuyor. Size mesela bir örnek vermem gerekirse bu lipidleri başka kompozisyonlardan oluşturabiliyorsunuz. Yani bu multivesiküler, çok katmanlı vesikülleri. Burada aslında aynı kompozisyondan oluşmuş ama boyadığımız boyalar farklı bu karışımı göstermek için. Şimdi sol tarafta görüyorsunuz ki daha birbirleriyle temas haline geçtiği anda birbirleri içinde yayılmaya başlıyorlar. Dediğim gibi bunu çözelti içindeki vesiküllerde, tek katmanlı vesiküllerde yapmanız çok zor. Çünkü yüzey üzerinde o kıvrılan kısımların aslında o bariyeri, enerji bariyerinin düşük olması ve karışmanın çok kolaylaşmasıyla olan bir olgu bu. Yani gözlem bu. Bunlar sonra tekrar aynı diğerleri gibi yayılıyorlar, yırtılıyorlar. Ve mesela şu uca baktığımızda, umarım mouse ikonumu cursor'ımı görebiliyorsunuz şu uçta. Ona baktığımızda oralardan da işte yine tüplerin oluştuğunu ve vesiküllerin çıktığını görüyoruz. Bunların kompozisyonel analizine, yani çeşitliliğine aslında baktığımızda da şöyle bir tablo görüyoruz. Şimdi bu grafik biraz karışık ama şöyle özetleyeceğim ben size. Burada gördüğünüz her veri noktası bir vesikülün, burada gördüğünüz bir vesikülün kompozisyonel olarak bir boyanın diğerine oranını gösteriyor. İşte şu tam kesikli çizginin olduğu yer birbirine karıştıkları yer. Buradan sağa doğru gittiğinizde pembe olan boyanın, magentanın sarıya oranı, sola doğru gittiğinizde sarının magentaya oranını görüyorsunuz. Ve burada gördüğünüz şey aslında kademeli olarak o karışma noktasından uzağa gidildikçe tek tek oranların iki tarafa da doğru yükseldiği bir çeşitlilik görmek aslında. Bunu dediğim gibi iki vesicle'ı sadece su içinde çarpıştırarak yapmak çok çok zor. Ben böyle bir deneysel bulgudan haberim yok açıkçası. Birinin diğerinden daha büyük olması halindeyse böyle bir bu hayatın başlangıcı bağlamında hep bu avcı işte yem olayı hep konuşuluyor. Çünkü bu işte seleksiyon ve işte birinin diğerine üstün olması olayı aslında deneysel olarak konuşulan, tartışılan bir şey. Burada da gördüğünüz işte bu sarı boyalı olan büyük hücrenin diğer hücreyi aslında bir anlamda yutması olayı. Burada da şöyle bir detay var. İşte bu tekrar birleşiyor, yırtılıyor ve buradan çıkan vesikülleri siz analiz ettiğinizde şöyle bir şey görüyorsunuz. Hani çok ilkel bir şekilde bunu fenotip ve genotip olarak tanımlarsak, yani böyle değil tabii temelde ama böyle tanımlarsak, yani fenotip hücrenin görünen özelliği, genotipte içinde saklanan ama göremediğimiz özelliği gibi düşünürsek aslında şu noktada baktığınızda, yani en sağ panelde veya aşağıki panellerde baktığınızda, yani bu noktada eğer deneye bakıyorsanız, daha öncesini bilmiyorsanız hiçbir zaman o magenta hücrenin burada olduğunu anlamıyorsunuz. Çünkü görünen bir fenotip yok ortada. Ama analizine baktığınızda eser miktarda o vesicle'ların içinde o magenta hücreden bir kısım olduğunu görüyorsunuz. Dolayısıyla aslında hani ata hücrelerden siz yavru hücre oluşturuyorsunuz ve o hücre, o ata hücrelerin özelliklerini taşıyor oluyor. Dolayısıyla bu deneysel sistem o açıdan da bayağı ilginç. Konuşmamın sonlarına geliyorum. Burada da size çok kısa şu anki çalışmalarımdan bahsetmek istiyorum. Şu an yüzey olarak yıldız tozu kullanıyoruz. Ben böyle bir şeyin varlığından da çok haberdar değildim. Yani bunlar aslında yıldız tozu popüler olarak o bağlamda söylenen ama işte mikro düzeydeki bunlar aslında göktaşları. Ve burada resmini, fotoğrafını da gördüğünüz Jon Larsen, bu işte Yıldız Tozu Nasıl Bulunur kitabının da yazarı. Aslında çok ilginç bir hikaye. Benim kendisi şu anda birlikte projede çalıştığım çalışma arkadaşım ve çok şanslı hissediyorum kendini. Şöyle ki aslında hani bilim ve araştırmacılara çok inspire edecek, ilham verecek bir hikaye. Çünkü kendisi bir çok ünlü Norveç'te bir caz müzisyeniyken ve aslında emekli olmuşken bir gün balkonunda yediği işte çilek tabağına düşen bir partikülün diğer toz parçalarından daha farklı olmasını fark edip onun ne olduğuna bakmakla başlayan bir macera aslında. Ve şu an aslında bütün çok yerleşik bilim enstitüleri, üniversitelerinde kabul eden bir protokolün gelişmesini sağlamış. Daha önce hep hani Antarktika'dan falan toplanıyormuş bu yıldız tozları ama şu an kendisi şehir ortamında çatılardan toplanan o tozların içinde bu yıldız tozlarının nasıl rafine edildiğini anlatan bir protokolü yazan geliştiren bir insan. Ve çok büyük bir yıldız tozu koleksiyonu var. Yani öyle ki bazı kategorilerde yüzlerce, binlercesi var ve hani NASA'ya baktığınızda toplamda mesela o kategoride 3-5 tane, 7 tane yıldız tozları olduğunu biliyorum. Ve bu yıldız tozunun burada fotoğraflarında gördüğünüz aslında diğer doğal partiküllerle bir karşılaştırmasını yapmak adına burada İsveç'te bir kumsaldan aldığımız bir kum yani numunesine yan yana koydum. Tabii aynı oranda büyütülmedikleri için çok adil davranmıyorum bu kum numunesine ama hani çok kabaca partiküllerin farkını görebilmeniz adına bunu koydum buraya. Çünkü bu mikro partiküllerin çok karakteristik bazı özellikleri var. Mesela çok yuvarlak bir yapıları var. Dokuları belli bir yapı. Mesela kumda bunu görmüyorsunuz. Kum partikülleri. Bunlar da bu arada magnetik. Yani ikisi de magnetik partiküller. Yani bir mıknatısa cevap veren partiküller. Ama kum tanecikleri böyle çok köşeli, keskin kısımları var ama mikro parçacıklar öyle değil, daha yuvarlaklar ve genelde atmosfere girdiklerinde yandıkları için bu metal kısımları böyle bir yerine doğru çekiliyor partikülün ve böyle sanki uydu gibi böyle küçük metal kısımlar oluyor üzerlerinde. Biz de şimdi bu yıldız tozlarıyla çalışıyoruz ve işte yine aynı deneyi yapıyoruz. Lipidleri üzerlerine koyduğumuzda hepsinde değil ama bazılarında kompozisyonuna bağlı olarak bu proto hücre gelişimini görüyoruz. Şimdi tam burada particle işaretli değil sınırları ama eğer mouse cursor'ımı görüyorsanız şu yuvarlak yapı partikülün olduğu yer, üzerinde büyüyen bu proto hücrelerde onun aslında kontörünü böyle onun çevresinden devam ediyorlar yani. Bu henüz basılmamış, şu an hala devam etmekte olan bir projeden size göstermek istedim ama paylaşım konusunda biraz hassas davranırsanız çok sevinirim çünkü henüz basılmadı. Sadece size bugün bir yapıyı gösterebildim ama yüzeyi değiştirirseniz veya lipid kompozisyonu değiştirirseniz bir sürü farklı yapıya ulaşabiliyorsunuz. Proto hücre yapısına ulaşabiliyorsunuz. Onu buradaki makalelerde okuyabilirsiniz. Bana da ulaşabilirsiniz sorularınız varsa. Ama hani eve götüreceğiniz mesaj benim konuşmamdan şu ki yüzeyler işin içine girince aslında çok basit varsayımlarla bu farklı hücre morfolojilerinin oluşumunu görebiliyorsunuz. Çok basit varsayımlarda şunlar, işte yüzeyiniz var, lipidleriniz var ve sulu bir ortam var. Bu üçü olduğu sürece bunlar kendi kendilerini oluşturan yapılar ve sadece hayatın başlangıcı değil, sentetik hücre mühendisliğinde de kullanabileceği potansiyeli olduğunu düşündüğüm yapılar. Sorularınızı merakla bekliyorum.
Öncelikle sunumunuz için çok teşekkür ediyoruz. Sizin adınıza tema vaktine fidan bağışında bulunduk. Bağış sertifikasını e-mail üzerinden ileteceğiz. Şimdi 10 dakikalık soru bölümüne geçiyoruz. Katılımcılarımızı sorularını sormaya davet ediyoruz.
We extend our sincere gratitude to Professor BİYOGEN for her presentation, and we made a donation of saplings to the Tema Foundation on her behalf. We will send a certificate via email, and we now have 5 minutes for questions, and we encourage our participants to ask any they may have.
Şimdi ben bir soru soracağım. Proto hücre oluşumları tamamlandığı noktada genetik bilgi akışının incelenebilmesi ve evrim dediğimiz sürecin başlayabilmesi için araştırmalarda ne gibi tekniklerin geliştirilmesi gerekiyor?
Bu zor bir soru. Yani şöyle önce ya bu konuda çalışan bir sürü arkadaşım var. Şimdi benim uzmanlık alanım o genetik fragmentlerin içine girdiği süreç değil ama işte yani olay aslında kendi kendine otonom olarak kendini sürdürebilen, çoğalabilen, özelliklerini koruyabilen ve evrimsel baskılara dayanabilen bir yapı oluşturmak aslında. Yani buna giden bir sürü yol olabilir. O yüzden şu an çok hani spesifike edemeyeceğim onu ama sondaki amaç bu ilkel yapıların içine giren bileşenlerle aslında kendi kendini devam ettirebilen, çevreyle etkileşimde bulunabilen ve survive edebilen yani hayatta kalabilen bir yapı oluşturmak. Dolayısıyla biz hani 4,5 milyar yıl öncesine gidip ne olduğunu göremiyorsak bugün laboratuvarda o süreci başlattığımızda tekrar o kompleks hücre yapılarına doğru evrilebilen bir şeyi görebiliyor muyuz? Aslında büyük soru bu ve cevaplanması da çok kolay bir soru değil.
Teşekkür ederiz, sağ olun.
Bir soru da ben sormak istiyorum ama birazcık kişisel bir soru gibi kaçabilir. Ben biraz daha konservat yapılarını araştırmayı seviyorum. Bu yapıların normalde membransız organeller şeklinde de ifade edildiğini duyuyorum ve şunu merak ediyorum. Sizce bu hücre oluşumunda modern hücreler gibi şu anda modern hücrelerimizde işlevler farklı bir şekilde gerçekleşiyor sonuç olarak organellerde. Ve vezikül yapıları bunun için ne kadar elverişli, yoksa konservat gibi yapıların daha mı elverişli bir şey oluşturduğunu görebiliriz burada?
Ya aslında bu da çok güzel bir soru. Bu biyokonservatlar veya şu anda sanırım biomolecular condensates diye de geçiyor. Aslında bunların hepsinin bugünkü modern hücre yapılarında bir karşılığı var. Yani mesela nanotüplerinde var. Hem hücre içinde nanotüpler var, hem hücreler arası gösterilen nanotüpler var. Biokondensate'ler de aynı şekilde. Yani şu an hatta çok büyük bir research alanı haline geldi bu. Yani bunların hepsi hücrelerde var. Şunu hatırlatmak isterim belki bu bağlamda. Bu işte endosimbiyogenesis teorisi ne bakarsak aslında bu ilkel yapıların bazıları, yani biz mesela mitokondriya bir zamanlar bir hücreymiş. Ve bu hücrenin içe alınmasıyla mitokondriya organeli olmuş. Dolayısıyla yani o yapıların hepsi sonradan biyoloji tarafından kullanılmış yapılar. O yüzden ben birinin diğerinden aslında daha geçerli olduğunu falan düşünmüyorum. Aslında hepsinin o bütünlük içinde bir anlamı olmuş bir süre içinde diye düşünüyorum. Dolayısıyla biokondensate de çok önemli bir aslında ilkel hücre modeli. O yüzden gayet ilginçler. Keşke hani 10 tane kolumuz ve aklımız olsa da hepsini aynı anda araştırabilsek ama artık onu sizlere bırakacağız demek ki.
Umarız. Teşekkür ederiz katılımınız için tekrardan.
Ben de çok teşekkür ederim. Sevgiler, Türkiye'ye selamlar.
Teşekkürler.
Teşekkürler.
Değerli konuklarımız, bir sonraki oturum için 10 dakika ara verilmiştir. İkinci oturumumuz saat 11.10'da başlayacaktır. Ayrıca chat kısmında paylaştığımız formu doldurarak gün sonunda gerçekleşecek çekilişimize katılabilirsiniz. Oturuma katıldığınızı belirtmek için de chat kısmında ve aralarda linklerden katılım formuna ulaşabilirsiniz.
Dear guests, a 10 minutes break has been given before the next session. Our second session will begin at 11:10. Additionally, you can participate in our giveaway that will take place at the end of the day by filling out the form shared in the chat. To confirm your attendance, you can access the participation form via links shared in the chat and during breaks.
Değerli misafirlerimiz, şimdi Sayın Profesör Job Bokhoven'a sentetik hayata yaklaşımlar konu sunumunu yapmak üzere davet ediyoruz.
Now, I want to invite Professor Job Bokhoven to present his talk on approaches towards synthetic life.
Okay, great. Thank you so much. You can see my screen and you can hear me, right?
Yes.
Yes, we can.
Yes.
Perfect. Okay, so Saturday morning, so thanks for being here. I'll be talking about our approach towards synthesis of life or synthetic life, and we take a systems chemistry approach. This is a little bit different than maybe what you're used to, classical synthetic biology approach, but I think the field of systems chemistry is getting ready to create the building blocks that we would need in order to synthesize life one day in the coming, say, 5 or 10 years. And so the deal with systems chemistry is that we are studying networks of interacting molecules. So what does that mean? Molecules that chemically react with each other, that undergo interactions with each other, either covalently by making new bonds or non-covalently through hydrogen bonding interactions, or molecules come together to organize into, for example, a protocell. And what systems chemistry does is have not necessarily clean systems, so hundreds of molecules interacting with each other, 100 different types of molecules interacting with each other, and then looking for emergent properties. So properties that you would not predict based on the individual components, but are really a result of these intricate networks. Now, what is not a better emergent property than life itself, right? What if you could mix 100 different types of molecules together and life would come emerging out of it? That would be my ultimate dream, right? I think it's a dream of many, many scientists. If you would do that, if you would make life from simple molecules, mix them in the right conditions, and life comes sprawling out of that, you could ask questions like, how simple can life be? What building blocks can I now take away, and can I still get life? And what are the minimal amount of building blocks that I need in order to synthesize life? And how complex are those molecules? I think that could be an approach towards understanding the origin of life as well. If you make life first and then you strip away everything that is needed, you keep everything that is needed, but just everything that is needed. You keep the minimal ingredients. Then you can ask questions like, well, were those molecules there? And if not, were there other variations of these molecules? So you can answer questions like, well, it must have happened here because here I had the right condition. And I think that is our approach towards answers regarding the origin of life question. And I think it's also an interesting question if you were to go to other planets and then say, like, well, if I want to look for other life forms on Venus or on Mars or even exoplanets, what minimal ingredients would I be looking for over there? So therefore, this is the reason why we are trying to build life from the bottom up using non-biological building blocks. We do that using molecular self-assembly. So this talk takes a little bit of a turn, and I explain a little bit self-assembly first and our contributions to that, and then I'll come back to this, how we make life from that. So the next 10 minutes, I'll talk a little bit about what we've been doing in the field of self-assembly. And this is, of course, to get towards synthetic cells, assembling synthetic cells. So molecular self-assembly is this idea that you can make molecules, design molecules to interact with each other and then form larger structures. And this is a peptide amphiphile as an example, but there's millions of examples out there. And arguably, there's not a better time to be in right now because we really understand the rules now with protein design and with DNA origami self-assembly. We can now create almost any object that you can think of. And here's just one simple example, but there's millions and millions out there. And this is a peptide amphiphile that organizes with other peptide amphiphils to form fibers that we beautifully imaged over here using cryo-electron microscopy. So we can make almost any structure we can think of, yet we cannot synthesize life. If we look at self-assembly in biology, the rules are not different. And this is somewhat frustrating, right? So the self-assembly that we do in the lab is not using different types of non-covalent interactions. It's not using some interactions that only biology has access to. We have access to the interactions too. And what we're looking over here is a cell that is assembled, and we're looking at the cytoskeleton. And the cytoskeleton is organized through proteins, and we can also design proteins to assemble. So what is then different between self-assembly in biology and self-assembly in the lab? And one of the critical differences we spotted is that while the rules to make structures are the same, the building blocks are the same, but biology regulates the function of these assemblies using chemical reactions. And that is something we are not very good at. So again, the assemblies that are formed and the way that they are assembled, the hydrogen bonding interactions, that's all the same. We can assemble fibers. But typically, and what we're looking at right now is the dynamics of the cytoskeleton in the cell. Those are regulated through chemical reactions, and that we don't fully understand how to do that yet. I'll give you a bit more specific example. And here's an example of a molecular assembly. And the molecular assembly, we could build something like this, but the function of this molecular assembly is regulated through a chemical reaction, and that's what we are trying to do. Now, the molecular assembly is called the calcium 2+ ATPase. And so the calcium 2+ ATPase is a molecular pump. It is pumping calcium from one side to the other side of a membrane, and membrane we can see over here. And so it's embedded in the membrane and pumping calcium from this side to that side. Now, it's doing that against the calcium gradient. So while you don't see this in this picture, the concentration of calcium here is higher than here, and it still works. So if you think about that, that shouldn't work in equilibrium. So we know that this is a non-equilibrium function. And indeed, the non-equilibrium function is pumping calcium from one side to the other, and that doesn't come for free, and therefore we need to invest energy. Now, we typically think, if we think about biology, and well, investing energy always comes from ATP, and we don't think about much about it, but this is exactly what I mean with the function of this pump is regulated through chemical reactions, and this is the chemical reaction. So what happens is that we've got an ATP binding site or a phosphorylating site over here. So first we bind calcium, and when that is done, this site can get phosphorylated. So ATP becomes ADP, it loses a phosphate group, and this is now located over here. And that changes the thermodynamically favored state of this protein. Suddenly, this is not the favorite state anymore. It would rather refold, pushing the calcium to the other side. And here it can let go, release the calcium. So the affinity for the calcium is lower here. It lets go. And only when it has let go, then it gets dephosphorylated again until we're in all the way on the right state. And this means we can now undergo a new cycle, right? So now we're back in the original state, and then we can undergo a new cycle. And it does this over and over and over and over again. So what I'm trying to say here is that molecular assemblies, particularly in biological context, they are regulated using chemical energies or chemical reactions. And so ATPase is a catalyst for the ATP hydrolysis, and it does so. And while it is doing so, it uses that energy to pump calcium from one side to the other. Now, this is what we're not very good at. So we were wondering, can we do this too? Can we understand the rules here? Can we get better at this, regulating molecular assemblies using chemical reactions? And not necessarily a pump. Not that I would love to build a pump one day, but just in general, this whole conceptual. And so I call that molecular self-assembly 2.0, in the sense that molecular self-assembly 1 was all about, or is still about, how can we design certain structures to form? How can we make sure that this, for example, a protein embeds into the membrane? Molecular self-assembly 2 is like, how can we now make sure that the protein does something using chemical reactions? You can also call it chemically fueled self-assembly. Yeah? We're using chemical fuels in order to drive our assemblies. So this is what we've been doing the last 15 years or so, is basically developing a new toolkit for self-assembly, and that's basically regulating molecular self-assemblies using chemical reactions. So we've been doing that to increase our understanding of supramolecular chemistry, systems chemistry, and basically get an idea of how far can we push this. We can develop materials that have functions that we don't see in living systems, we don't see in synthetic systems, but typically see in living systems, like memory, adaptation, and self-healing. And finally, we can increase our understanding of life, its origin, and biology. And this is, of course, what this talk will focus on. I need to talk a little bit about how we now make these types of assemblies, and then we move on to our efforts to synthesize life. So we were thinking, okay, we want to regulate our molecular assembly process using chemical reactions. And so for that, we need to fulfill those types of requirements that we can actually do that. So now if we go back to that ATPase example, this is effectively what it's doing. On the left, we've got a catalyst, and the catalyst is converting fuel, ATP, into waste ADP and inorganic phosphate. And it does so by temporarily activating the catalyst, and that activated catalyst, as I said, has a different equilibrium position, so it refolds and then pushes the calcium to the other side. That's what we're looking for. Can we activate a catalyst, deactivate the catalyst, activate the catalyst? If you want to do that, you need a catalyst that is, on the one hand, converting fuel to waste. And you need, of course, a fuel that is has a high chemical potential in order to drive this reaction, also in order to undergo a change of the activated catalyst. That's our ATP, of course, in biology. And that should have a high energy barrier. That means that the fuel doesn't spontaneously combust or doesn't spontaneously fall apart, but it needs that catalyst in order to do so. And you want to avoid any types of side reactions, because anytime your catalyst makes a side reaction, it cannot be used anymore, and that will be disastrous. For example, for biology, if a pump every 5 reactions, it does a side reaction and then cannot be used anymore, it means that the pump is pretty useless. It needs to do millions and millions of reactions in order to do this. If you prefer energy landscapes, this is what we're looking at. The fuel needs to have high chemical potential. So this is all released, by which we can, for example, pump calcium from one side to the other. But it also needs to have a high energy barrier so that it doesn't spontaneously combust. And ATP is an amazing fuel from that point of view. It hardly hydrolyzes because of those 3 negative charges. But you need a catalyst in order to liberate that fuel. And that's what we have on the right. We see the catalyst gets activated, liberates the part of the fuel, and then gets deactivated and can undergo cycles and cycles and cycles. Okay. So we wanted to build that in the lab. Once again, systems chemistry, synthetic life from the bottom up. So we don't want to deal with ATP. We don't want to deal with enzymes. We want to use this principle of making molecules out of equilibrium and then letting them do some sort of an assembly type, but not using chemical fuels from biology, not using enzymes from biology, because I don't know if you can answer, if you produce synthetic life with biological molecules or based on biological principles, I'm not sure if you can answer many questions related to the origin of life question. So we're mostly interested in, can we do this using simple molecules that were never part of a biological entity? And so this is the reaction we came with, and it is also one of the few slides with chemistry in there. So bear with me. What we have on the left in blue is this catalyst, and that's our catalyst to liberate energy from our fuel, the purple molecule, which is called a carbodiamide. And carbodiamide are used as coupling agents, condensing agents in peptide synthesis, as well as labeling your protein. So it's a very popular molecule. And we thought that was important. Let's take a popular molecule that many people can work with this reaction cycle. And so we choose this carbodiamide in most biochemical labs and in most organic chemistry labs. These molecules are available. We even see this in the biophysics labs. And this, of course, is a bis carboxylic acid, has 2 carboxylic acid, a catalyst, and this is part of an aspartic acid group, so it could be a C-terminal aspartic acid. Now, if you combine those two, you, in a 2-step process, activate your carboxylic acid into what is called the activated catalyst, which is an anhydride. And we do that by converting a molecule, the carbodiamide, into its corresponding urea. Literally, we've put on a molecule of water onto the carbodiamide. So if you go from the carbodiamide to the urea, you see there's now H2O on there. And the H2O came from this catalyst, one of the oxygens and 2 protons, making the anhydride. Now, the anhydride in water is particularly unstable, and that's exactly what we want. We want this to hydrolyze quickly. It reacts again with water, splits up, and is now back in the catalyst state. So we have a reaction cycle that is activating our catalyst temporarily. Think of that as the phosphorylated species. And this spontaneously dephosphorylates, or in this case, hydrolyzes. Now, it's important to realize on the left, you have your catalyst. On the right, you have your activated catalyst. And the activated catalyst is only alive for about a minute. So it's a half-life of a minute. So if you mix everything together, you start making these anhydrides that are on for about a minute and then are switched on, statistically 50% chance that it gets switched off again. And it's basically blinking on, off, on, off, on, off until the whole system has run out of fuel. It is a popular reaction cycle. There's now many other labs that are using this chemical reaction cycle, which I think is pretty cool. It's also nice that people can also work with this cycle. And people are using this, on the one hand, to operate their molecular machines. So we see molecular motors to drive endergonic synthesis and many other examples. But let's focus on our work specifically, and then we move on to this synthetic life. How we're using this chemical reaction cycle in our approach towards synthetic life. Now, one of the things we do is we use this loss of these 2 negative charges on the catalyst. We're activating the catalyst, it gets deactivated, activated, deactivated. We use this loss of those 2 negative charges in order to drive our self-assembly. So if we go from left to right, we see that the catalyst that is activated, remember it's only there for about a minute, has lost the 2 negative charges. And because it has lost those 2 negative charges, we can now use design rules in order to make this molecule assemble, which will become later important for this synthetic life part. So we activate it, it can now float around, zoom around for about a minute, and then it gets deactivated. If it finds in that minute another molecule to assemble with, it can assemble into a stack. We design it in that way because it has hydrogen bonding interactions between the amino acids, as well as pi-pi interactions between the phenylalanine. And that makes it assemble into a stack, for example. So that works. What we then see is one principle, and the one principle we get is that if you do this, we look at these images, we get it from the webcams. If you do this, all of the materials that you get, in this case, we get a hydrogel, all of the materials that you get are regulated through chemically fueled processes. And that means that all of these materials are only temporarily active. So just like your car, if you throw a bunch of fuel in your car, you can only drive a bunch of kilometers. In our case, if you throw a bunch of fuel at your solution over here, you get a hydrogel, but that hydrogel is only temporarily active. And for how long it is active depends on how much fuel you added. So if you add a lot of fuel, you get a gel that is alive or active a bit longer compared to if you add a little bit of fuel. So that's what that looks like. We have a hydrogel with 1, 2.5, 5, 7, or 10 units of fuel. And as you see, the less fuel you add, the shorter this material is alive. This works for gels, this works for inks, this works for droplets, this works for any kind of materials. As long as you have a bunch of fuel, so in the middle graph, the material properties against times, you shoot in a bunch of fuel. The system is consuming the fuel. It brings the system out of equilibrium. But the moment we set back equilibrium, the material is gone again. As long as energy is available, the system is out of equilibrium, giving our material. And after that is game over and the material is gone. And so that's what we did in the early years. And then we was like, wait, what if we now conceptually put that onto synthetic cells? So what we've done is we've stolen a concept from biology, which also regulates stuff out of equilibrium. And then what is this ultimate thing that is out of equilibrium? That's life itself, right? So doesn't life do something similar conceptually? I mean, obviously, this has nothing to do with life synthetic or self-erasing inks or hydrogels. But conceptually, there are similarities. Life can only exist in equilibrium. The moment it runs out of energy, the moment that you stop eating or you stop feeding your bacteria, it's game over, right? So they can store that energy somehow, but at some point, the system needs to be brought out of equilibrium, because when equilibrium sets in, it's game over for the bacteria. So what if this, rather than material properties, would be synthetic cells, right? What if we bring our synthetic cells, we design them in such a way that they can only be made when there's fuel present, but the moment the fuel is gone, it's game over. At least now we have this idea of selection pressure, right? If you get a lot of fuel inside of you, you might live a little bit longer than your competition who got only a very little bit of fuel. So that was an idea we'd been playing with and have been playing with for the last couple of years, and the rest of the talk will focus on that. So again, we took this concept from biology and then applied it to systems chemistry to create stuff out of equilibrium. And now we're wondering, can we do this in order to synthesize life? So we got together with about 50 scientists in 2023. We organized a workshop together with Christoph Weber and Kerstin Göpfrich. And we sat down and we talked about how could we synthesize life? If we want to build life from the bottom up, what do we then need to do? And first of all, sadly, you do need then some sort of a goal, of course. You can't be saying, I'm going to synthesize life without a goal. And if you want a goal, you need a definition of life. And that's why I said sadly, because if you start defining life, everybody has an opinion. And none of these opinions is the same. But since we do need a goal, we said that, well, at least let's say, well, what is the overarching goal? And therefore, how do we define life? Maybe this is the most minimal, simple definition of life. But that would be the easiest goal. And then from there on, we can build and make more complex life forms. So we basically, inspired by NASA's definition of life, a self-sustaining chemical system capable of Darwinian evolution. We prefer to think of synthetic cells. So we basically said a self-sustaining compartment that is also capable of Darwinian evolution. And there's that idea already of self-sustainment, right? So this is a compartment that needs to keep on sustaining itself. It needs to keep on avoiding equilibrium through metabolism. It basically keeps on building molecules in order to keep itself in its state, right? And so we were thinking of a box over here, and the box is basically sealed off except from here matter flows in and out and energy flows in and out. And the box needs to be kept out of equilibrium because we know life cannot exist in equilibrium. And inside the box, we have synthetic cells, and they're sustaining themselves. So that basically means there's a metabolic reaction network inside of it that keeps on producing the molecules in order to keep itself in that state. Now, what state does it need to be? It needs to be in a state in which it can undergo Darwinian evolution. Yeah, so it needs the minimal amount of ingredients. If we want to make a minimal life form, we need a minimal metabolic network that keeps itself in a state that can undergo Darwinian evolution. And so what is Darwinian evolution then in such a context? It's basically that state needs to, from time to time, make a copy of itself, it needs to replicate itself, and not a perfect copy. It needs to make mistakes on purpose. It needs to mutate. Because only through mutations, we have synthetic cells that are slightly different. And because they are slightly different, we can now apply this idea of selection pressure or natural selection. And so that means that the weaker ones will die off. Again, they will get closer to equilibrium and decay off. They can't replicate themselves anymore, whereas the stronger ones can. So the stronger ones take up the resources that were available for the weaker ones, and that is the concept of evolution. That will be Darwinian evolution. So that means if you have 4 replicators and replicator A, B, C, and D, and sometimes A wins under certain conditions, B wins, and A mutates into B, that will be Darwinian evolution. We then say, like, well, actually what you need is open-ended evolution. Open-ended evolution means there's A, B, C, D, but the moment that you've discovered how to make D, it opens further. The genotype space opens further. Then suddenly E, F, G, and H become available. And then when you figured out how to make H, then the whole alphabet's available. And this keeps on going on and on and on. That means open-ended. There is no end to this evolution. That's literally, when I'm looking outside right now, that's what I see outside, right? There's not one genotype. There's a million genotypes. And the moment you figured out another genotype, then evolution goes further on that. So if we were to go to Mars one day, then I'm pretty sure there will be a new genotype opening up over there, or what we still see is new species are emerging every day. There's this idea of open-ended evolution. So we need more than 4 replicators. We need this idea that this can keep on going on and on and on and on forever. If you synthesize that, then we could say, well, I think you have synthesized life, at least to NASA's minimal definition. Now, people may disagree with that. This is as I said, this is the minimal definition. You should also not get too stuck on definitions and semantics, but this is essentially what life is doing. And from this, you can expect that life would go on and on and on. And then at some point, you could end up in a situation where we find ourselves now with biological life. Okay, so again, we need a synthetic cell that is somehow keeps itself out of equilibrium or is kept out of equilibrium, makes copies of itself, mutates from time to time, and then selection pressure selects the fittest mutants. So we were thinking, what if we use our chemical reaction network that produces assemblies out of equilibrium and use that as a sort of a selection pressure? If we now fuel such a container or such a box or such a reactor from time to time, you can expect that assemblies emerge. They're competing with each other because there's only a limited amount of fuel left. And again, the fuel is being irreversibly consumed. So that's being degraded. And as the fuel is depleted, the compartments will decay. But if you now fuel it at the right time, you wait long enough so that most of the compartments have depleted except for a few, those that have survived have a selective advantage because they received a new shot of fuel. They can take up building blocks quickly because there's this beautiful rule that really helps us. And the beautiful rule says any assembly or any assembly that is formed through nucleation process, it is always easier to grow something that is available as opposed to nucleating a new one. So it is easier to make to grow a compartment that already exists as opposed to growing or making a new vesicle or droplet or whatever we're dealing with. So that means that the new shot of fuel will predominantly go to the assemblies that still existed. They can grow, whereas the ones that have died off are gone. So we tried this concept. So like, okay, well, let's build chemically fueled synthetic cells. So synthetic cells that require fuel in order to stay alive. We did that with oil droplets. We did that with vesicles and block-polymer micelles. We did that with oil droplets that are extremely simple, made of molecules that you can find on asteroids. We're now working on vesicles that are also extremely simple aliphatic tails, carboxylic acids. We've done it with coacervate-based droplets. Coacervate-based droplets are a bit more complex, a bit less origin of life-y, but I do like to talk about them because they have this beautiful, beautiful advantage. And the beautiful advantage is coacervate-based droplets. I think my Turkish, as I said, is a bit rusty, but I think I heard the previous speaker talk about biomolecular condensates. That's the same principle over here. The beauty of them is that they have plenty of water inside of them. And we can use that water to do all kinds of chemical reactions. So I'll talk about the biomolecular condensates or our complex coacervate-based droplets. And so this is what that looks like. We have our peptide on the left, on the top. And this peptide is in its catalyst state. That means it's not activated, and it was designed not to do anything. It doesn't interact with any polyanions. It doesn't make any droplets. It doesn't do anything. We can now chemically activate it. Remember our reaction cycle. We get rid of the catalyst. The blue part of the molecule is now converted into the anhydride. And in that anhydride state, it is now activated. But this anhydride state only lives for about a minute. Yeah? That was the half-life of a minute, and then it gets killed back there. So that means that when you add fuel, you have a molecule that goes from 3 plus charges and 2 minus charges. So it's zwitterionic plus 1 to fully 3 times positively charged. So zwitterionic plus 1, 3. Zwitterionic plus 1, 3. And it does that. It blinks from on, off, on, off, on, off. It does that for as long as there's fuel present. The moment the fuel is gone, it's game over. The system falls back into this state. So think again about your car. You give it fuel, it can drive a couple of kilometers. In this case, it can produce these molecules for some time, and then it's game over. If we now mix in a polyanion like RNA, DNA, or synthetic polyanions, what happens is that if you have the solution at the bottom, your molecule is not interacting with that RNA until it gets chemically activated. Now it is alive for a minute, I said, half-life of a minute. It's now zooming around looking for RNA. It binds the RNA, and that forms some sort of a complex, right? So an RNA bound to this cationic molecules binds other RNA and forms what is called this complex coacervate droplet. That's basically a spaghetti bowl of RNA molecules held together by these cations. But again, the cations are transient. They are not there forever. They're only there for a minute. So 50% of this of the droplet's peptide is being deactivated every minute, and it cannot do anything there. It doesn't interact with the RNA, so it has to leave again, get reactivated. And it's insane when you think about it. You have a droplet there, 50% of its material needs to be kicked out of the droplet every minute. 50% is being replenished by stuff from the outside. So it's a very, very dynamic environment, and the droplet can only be there for as long as there's fuel present. So we played around with a couple of iterations of this molecule, and eventually we dumped it in front of a webcam, and eventually a molecule worked. And I was super excited to see that. We have a solution that is clear, a solution that gets turbid, and then clear again. And that was for us an indication, okay, something happened, there is something there, and it's only there for 18 minutes. Poor little droplets, they only have a life of 18 minutes, and then it's game over again. Huh? And so we go to the microscope, and we indeed see spherical things. And that looks like droplets. And what really excited me over here is that the lifetime of the droplet very nicely matches, so the turbidity very nicely matches the product that we measure in the HPLCs. So we have a very, very tight correlation between the number of molecules that are activated and the total turbidity of our solution. Droplets emerge in response to fuel. They sustain themselves by taking up material from the outside, but then they decay eventually if it's game over. If there's no more material coming from the outside, it's game over. So we teamed up with the lab of Kerstin Guffrich, and with her expertise, we built microfluidic reactors. And in the microfluidic reactors, the precursor, our catalyst, our fuel, and our polyanion is all mixed. And then we trap that into a microreactor. So that's a droplet like over here. We trap it in a microreactor, and then we can image the whole process. The birth, the emergence, the sustainment, and the death of our active droplets. And so this droplet here that we have on the center of the slide, that was produced about 500 milliseconds ago. And then what you can see is that we see the birth of our droplets. We see that they're zooming around. They fuse a lot. Some of them sink to the bottom of the reactor. That's why they're lying still over here. They fuse a lot, and eventually it's game over 12 minutes later. Yeah. So we can now track those droplets. We can look at them individually. We can say things about them. And this is, for me, the point that I really start thinking we're building synthetic life, right? You get emotionally attached to them. You give them names. I call this one Jimmy over here. It's the big one. Jimmy just ate his neighbors. That's why he's so nice and fat. But eventually, it's also game over for Jimmy. And we are back in equilibrium again. And everything is gone. Yeah. So the life of an active droplet is very, very short. And it only lives for 14 minutes. And in those 14 minutes, it shows all kinds of cool behavior. As I said, they love fusing. They have a very low interfacial surface tension. So they grow through fusing, and they do that very, very quickly. And we can see that in the number of droplets over here. The number of droplets goes down as a function of time. They show this non-equilibrium dissolution process, which is basically the core is depleting before the shell. And that basically molecules are still entering this droplet as they are depleting. So they're decaying right now. And the droplet is still receiving droplet material from the outside, but the core is just depleting faster than the shell can keep up with, or the outside solution can keep up with. So we go from a bowling ball that is completely consistent inside into a soccer ball, basically a shell of droplet material, until it's game over. And then finally, the last seconds of such a droplet, count with me, we've got 1, 2, 3, 4 droplets on this slide. And finally, the droplets decay, not as what I expected. They rip apart into tiny droplet fragments. And that's, of course, super exciting because it is, well, not self-division, but it is a mechanism by which, what did I say? Count with me, 4, I think. 4 droplets became, I'm not going to ask you to count this, but it's more than 4. So not a beautiful self-division mechanism. And if you would ask me which mechanism do you prefer, obviously we want droplets to split nicely in 2. But it is a mechanism by which you could think, well, we're producing offspring, right? There is 100 droplets now. So maybe that's some sort of an offspring or decay mechanism. So we decided what's actually happening in that decay mechanism? Why does it do that? And we were a bit naive and a bit dumb. And so what we did is we bought polyanions, RNA, as I said, and we bought poly-U from Sigma Aldrich. So poly-U is basically this uracil chain. And we thought we would buy monodispersed poly-U, but we didn't. We bought poly-U that has a very, very broad polydispersity. And a very, very broad polydispersity meant that while we thought we worked with a monodispersed poly-U, we had many different chain lengths in our droplets. And so we started thinking, well, maybe that's responsible for the self-division mechanism. So our mistake was actually turned out to be a good one. So Monica started figuring out, okay, what's going on exactly over here. So what she did is she, instead of buying polydispersed poly-U, deliberately made long, we're now dealing with another polyanion, PSS, polystyrene sulfonate, and short PSS, mixed those together to make droplets. And she found that both of them make droplets just fine. But what you then see is that the long one tends to separate from the short one within the droplets. So it starts making droplets within droplets or speckles within the droplets. And so the scheme kind of shows that the long purple polyanions start accumulating into this kind of beads. And those beads, because they're made of long polyanion, they're a bit more difficult to dissolve. And because they're a bit more difficult to dissolve, they survive slightly longer than mama. And so as mama or as Jimmy is dissolving over here, what we see is 1, 2, 3, 4, 5, 6, 7 speckles. And she's really, really cool. She can really control this process nicely because she can control it nicely. First of all, because she understands what's going on, she can now control it. And so depending on the fraction of this long PSS and this short PSS, she can control how many offspring each droplet will produce and for how long the offspring will live. And now you think, well, the offspring will live for what, 60, 80 seconds? But keep in mind that the mother also only lives for, what did I say, 12 minutes or so. So we have 12 minutes of mama, then mama rips apart into 12 or 18 offspring droplets, and those live another 60 seconds, and then the offspring also dissolves. So keep in mind, once again, the offspring is held together by the same polycation that eventually dissolves. So everything goes to equilibrium. There's nothing left of the mother, but also nothing left of the offspring anymore. So we can control the offspring number, and we understand this mechanism. So let's go back to our wish list. We wanted self-sustaining compartment that harvests nutrients from the environment. So we've got a compartment that is indeed doing that. It's getting materials from the outside. It is a non-equilibrium compartment, and that's the thing that I'm really pushing over here. If you want to make life, you need to think of some sort of a non-equilibrium decay mechanism because otherwise there is no selection pressure. If everybody just lives happily after, including your synthetic cells, there is no selection pressure, and therefore there is no evolution. At some point, all the resources will be consumed. Everybody's happy. There's no competition. We can't do Darwinian evolution in equilibrium. So we need to think of death mechanisms, decay mechanisms, selection pressure, and ours is basically couple it to a chemically fueled reaction. And for as long as there's fuel present, there will be pressure in order to do well and make it to the next shot of fuel. And so that's what I want to go next to. It's like, can we now do that? Can we rescue droplets with a second shot of fuel? Because it's nice that you have cycle 1, emergence, fusion, starvation, division. But if that's it, we're not going to do evolution. What we need to do is make sure that we can save those daughters or save the ones that didn't produce daughters by shooting in a second batch of fuel. And this is super duper complicated. So we built another microfluidic device. With the other microfluidic device, we can now open a chamber to refuel our droplets at exactly the right moment. And so we're currently writing up this work. This is what that looks like. These are the first experiments in that direction. What we see over here is a droplet in the center, the blue one, and it is about to divide. So these are Monica's recipe, a new recipe on how to make it divide. It rips apart into plenty of daughter droplets that just happened. And that is exactly the moment where we reopen the chamber again and see how it grows again. Now, it doesn't grow nicely as we expect, just ballooning up again. It takes up material from outside. It also bumps into other droplets and then lives on again. But at least a part of a fragment of the original mother, of the original divided droplet, it divides, the fragments are going left and right. Part of that fragment recombines and now is the next generation droplet again. And we can repeat that, follow the droplet and go for the next division round. Somehow it changes color here, boom, and do another division route and grow again, and do another division route and grow again. So what we have now is a mechanism by which we can capture, can save, rescue our non-equilibrium droplets by opening up this chamber again, letting new fuel in, and save it for the next generation. And rather than living for 11 minutes, is now there for about half an hour. And then at one point, if you forget feeding your synthetic cells, it's also game over for those. So then they rip apart one last time, but this time we did not open the valve again, and therefore the daughter droplets overlived. So this is what that looked like if you now look at the entire microfluidic reactor chamber rather than one droplet. What you see is the number of droplets that has emerged is about 100. It drops rapidly because of starvation, because of dissolution, and because of fusion. Division sets in around 10 minutes. I should not say division, it's not really classical division, it's more like offspring production sets in. And as we said, 70 droplets become about 200. So on average, each one produces 3 fragments that rapidly, now we open a chamber, we've saved those fragments, rapidly they bump into each other, dissolve, and fuse again, so that number declines again really, really quickly, and they live on. And they do that again, and now we fuel them again. And they do that again, and now we fuel them again. So now for 4 shots, we can save our droplets. And with each shot, we have a shot of fuel, over here. And with each shot, we saw the division event, so an increase in the number of droplets. And then eventually, as I said, this is a non-equilibrium, chemically fueled droplet system. That is important. They're all competing for fuel. And those that have dissolved, it's game over, right? It's game over for them. They can't come back because the competition that lived on gets the new shot of fuel and nucleates quickly into new droplets. Sorry, new droplets cannot nucleate, but instead all droplets rapidly grow until eventually equilibrium sets in and all droplets are gone. Okay, so we have this competition mechanism. This is not self-replication, but we've got this division mechanism, and we also have a mechanism by which one offspring survives and another one decays off. But there is obviously no genotype. There's nothing that says you are Jimmy and you are Timmy, and somehow Jimmy is stronger than Timmy because of what is it in life as we know it. That's of course the genotype, right? The fact that our DNA encodes for our success, the genotype encodes for the success of the phenotype, which is then going back to the genotype again. So that's what we're now looking for, of course, is a molecule, some sort of a molecular system within each droplet that makes that droplet unique. In our bodies, that's the genome. In a synthetic droplet, that's what we need to design right now. So that is a molecule that is influencing the success of the synthetic cell, the phenotype, that is passed on to the next generation and contains some sort of an information, and the information encodes, of course, for the success. And so that it would be a genotype that needs to be able to replicate itself. As the thing is dividing, it needs to replicate itself. That is passed on to the next generation. If Jimmy produces offspring, you make sure that everyone has the same information-carrying molecule as Jimmy, and that it doesn't get diluted. So if you do that, you say, well, I'll take a lot of genotype molecules. It gets diluted with every generation. So you need to make sure that you make as many genotype copies as you are producing offspring. And that's a lot. Each droplet can produce up to 12 offspring. So we need a molecule that replicates at least so that there's 12 of them over the relatively short lifetime of the droplets. And then that genotype needs to influence the phenotype. It needs to increase the success. So if you have this molecule inside of you, you live longer compared to somebody who has a different molecules inside of them. And so in that way, the genotype lives on and is successful. So that's what the genotype does. The genotype helps the phenotype so that the phenotype helps the genotype. So effectively, the only thing the genotype does is help itself through this idea of this phenotype. So that got us thinking, okay, so what if we can build such a thing? We need a reaction, a chemical reaction that somehow produces another molecule of itself that gets passed on to the next generation in case a droplet splits in 2 or 3 or 12 droplets. So we need an autocatalytic, even better, we need a self-replicating molecule. So we thought of, this is difficult because our chemistry is a bit nasty. It involves electrophiles and nucleophiles. So we need to have specific chemistry that doesn't deal with electrophiles and nucleophiles. Otherwise, you get all of this crosstalk. So we came up with, Hector came up with a click reaction. And this is based on work by Devaraj and Whitesides. Sergei Semenov, who's in Israel right now, developed this type of chemistry. And basically what this means is we've got a click reaction. Now, I'm not sure if you know about the click reaction, but the click reaction is basically a reaction that is simple, fast, gives us almost quantitative yields, and is practically irreversible. And so what we do here is, I don't know if you know this specific click reaction, if you've ever done the alkyne azide cycloaddition reaction, you may have needed to buy a catalyst, and the catalyst you can buy from Sigma-Aldrich is this molecule here, and that itself is a product of the click reaction. So this reaction is in fact autocatalytic. So this molecule here clicks onto that one arm, a second arm, a third arm, and the reason why it's autocatalytic is because this strongly binds copper 1+, prevents it from oxidation, and because it strongly binds copper 1+, it can catalyze this chemical reaction. So if you start with these conditions, the reaction is extremely slow. I can actually show that. The reaction is extremely slow. But the moment that you start producing this autocatalyst, this catalyst here, the product, the moment you first make the product, then it starts accelerating until it depletes all of the building blocks. So it's an autocatalytic chemical reaction that gets faster and faster as it is progressing. Now, that's great. Can we now use that in the context of our droplets? So what we can see on the one hand, with the droplets or without the droplets, the reaction is equally fast. So that's great. It is compatible with the droplets. But the problem then is autocatalysts are not replicators. It is conceptually important. An autocatalyst produces itself, a replicator produces itself and only itself. So an autocatalyst will produce any kind of chemical reaction that involves an azide and an alkyne, whereas a replicator will not. It's basically the equivalent of DNA will only make a copy of itself versus DNA making any kinds of DNA bonds with untemplated, randomly starting making nucleotides. Making oligonucleotides. And that's exactly what we're dealing with, an autocatalyst. So it doesn't care. If I give it 2 types of azides, it will convert them both equally. And it will get a more or less statistical distribution of the products. I draw them like this, the alkyne in the center. And we have the 3 azides over there. So it makes statistical distribution of the 3 products. So autocatalysts will catalyze each other's production. So that's not a good replicator. Or you could think of it as a replicator that constantly makes mistakes, a horrible replicator with a very low fidelity. However, if we now do that in droplets, droplets can make autocatalysts replicators. They're not great replicators, but they can lean into replicators. What does that look like? If we have this replicator here, we give a little bit of that and we put that inside of the droplets. What we see is that if we give it a mixture of product, we don't get a statistical distribution anymore, we get more of this product. So it is producing more of itself. And the reason why it is producing more of itself is because it likes to partition in the droplet, and so does its fellow azides like to partition in the droplet. But the other azide doesn't like to partition in the droplet. So it selectively sees more of this azide than that azide, because this one is negatively charged and this one is positively charged, and our droplets love taking up positively charged stuff. So the replicator goes in the droplet, and so does its building blocks, and therefore we get a decent replicator, decent with a big asterisk. The phenotype, the droplet, helps the genotype to start leaning towards more production of itself. Now, the genotype also helps the phenotype. So again, the droplet helps the replicator to form an autocatalyst to become a replicator, becomes more selective, start producing more of itself. The genotype also, reciprocally, helps the phenotype. And why? Because it's a positively charged molecule. And remember, our droplets are decaying because they are losing their positively charged molecules. So if you now start putting in their positively charged molecules, you help the droplet. You give it cationic molecules, you're starting to help it. So while it is decaying, this replicator is kind of holding together the last RNAs. And no, don't leave, I'm still here. So the decay is much slower. So this is a molecule, this is a droplet. The gray trace is a droplet without genotype, the control, and that decays over the course of 5-6 minutes. Whereas the one with the genotype lives much longer. And this is what that looks like. The one on top is with the genotype, and the one at the bottom is without. As you can see, after 10 minutes, it's completely game over. Whereas the one with the replicator inside of it lives a little bit longer, gets a different morphology, and eventually actually lives for like a good 40-50 minutes. So the lifetime can be extended by having a genotype inside if you, provided that it is actually partitioning inside of you. The ones that don't partition inside don't help the droplet. And what I thought was really, really cool is that this trick only works, the genotype only affects the phenotype, if the genotype is made inside of the droplet. So if you first make the replicator in solution, then you fuel the droplet. So look at the experiment on the left. We first make the replicator, then we give it fuel to make the droplets. Then the droplets don't have time to pick up the replicator. So the replicator wants to go in the droplets, but it's all so fast, they don't have time for that. So the genotype must be produced inside of the phenotype in order to affect it, otherwise it doesn't work. And this is important. This is important because if the droplet were to die, it loses its genotype. This genotype can now not start infecting other droplets. They don't help the other droplets, they don't kill the other droplets, they don't do anything because they don't have time for that. So this only works, the genotype must be produced in the phenotype in order to affect it. And then finally, we did one last experiment, and then I also stopped talking. And the last experiment is, does the genotype get passed on to the next generation? So we fuel our genotype and our droplets, and we see that indeed these droplets live longer. But if we let them refueling, if we let the refueling happen every 8 minutes, then we can save this droplet, and we can save it again. And what we see is that indeed then the genotype benefits, the phenotype benefits from the presence of the genotype. That means if we seed it every 8 minutes on the bottom left, the droplets live slightly longer compared to a control experiment, but also compared to if we only seed it every 90 minutes. So we start with the seed and now we fuel it every 90 minutes. Every time we do this, the droplet loses its genotype, needs to start over again, and doesn't have this benefit that the ones have that live on. So in other words, you need to be surviving in order for your genotype to stay inside. If you somehow died and start over again, you don't have this benefit anymore because you've lost your genotype. So the genotype is passed on to the next, I don't want to say generation because there's no division. This is the equivalent of having breakfast, lunch, and dinner. So basically, the droplet survives because it keeps on eating. And because it survives, the genotype stays within that droplet, and the benefit also stays within the droplet. If it were to decay somewhere halfway, it forgot to have lunch one day, and it dies off, then it's game over, and it has lost its genotype now. Okay, and with that, we kind of demonstrate this reciprocal coupling between genotype and phenotype. The genotype, the replicator, helps the phenotype, obviously, by increasing its longevity. The droplets live longer. And because it lives longer, it manages to survive a starvation route, and therefore this helps the genotype again, the replicator, because it helps the replication process. So the genotype helps the phenotype, which then helps the genotype. And this is the whole idea of this selfish gene, a book by Richard Dawkins. And that's the idea that basically the genes don't care, right? The DNA doesn't care about you living long or living short. The only thing that DNA cares about is, am I being propagated? And it's the same over here. The genotype itself doesn't particularly care that the phenotype lives longer, but it cares about the fact that that is more successful for its own replication. So we're setting steps towards this idea of synthetic life, but the one thing we're clearly missing is mutations and different genotypes competing with each other. All the experiments I showed is one genotype, and I compare that to molecules or droplets without a genotype. What we now need is genotype A competing with genotype B, and then we can start doing these kind of natural selection experiments where I hope to see that genotype A under certain conditions lives longer or replicates better, and under other conditions it's genotype B. Okay. Yeah, we also work with synthetic cells. I don't want to waste too much time on that. As I said, this is a movie from Synthetic Cell. I do want to show it because I think it's an amazing movie where we see synthetic cells that can also self-divide, but I'm not going to talk into too much detail about this work. But we have other types of synthetic cells in the lab as well. So with that, I would like to stop talking. What have we learned? We are building chemically fueled self-assembly, and then specifically in order to create synthetic life. And our approach to that is to build self-sustaining compartments that need fuel to survive. And I think that's critically important to understand from our work. Chemically fuel is required in order to stay alive. Without it, it dies off. So there's a selection pressure to survive until the next fueling round. These self-sustaining droplets, they can self-divide. Autocatalytic molecules can become replicated within them. So we give them a genotype in the sense of an autocatalytic molecule. And those autocatalytic molecules are passed on to the next generation after division, as well as after fueling rounds. And that affects the behavior of the droplets, which then reciprocally affects again the behavior of the replicator. We would like to build mutational replicators, of course, in the future, adaptation, and then demonstrate Darwinian evolution in these kind of things. With that, thank you very much for your attention, and thanks a lot to my team as well as funding agencies. And if there's any questions, I'd be happy to answer those.
Sayın Profesör Job, bu konu konuşması için teşekkür ediyoruz. Kendileri adına tema vakfına fidan bağışında bulunduk. Bağış sertifikasını e-mail üzerinden ileteceğiz. Şimdi 5 dakikalık soru bölümüne geçiyoruz. Katılımcılarımızı sorularını sormaya davet ederiz.
We extend our sincere gratitude to Professor Job Bokhoven for his presentation. We made a donation of saplings to the Turkish Foundation for Combating Erosion, Reforestation, and Protection of Natural Habitats Foundation on their behalf. We will send the certificate via email, and we now have 5 minutes for questions, and we encourage our participants to ask any they may have.
Hello, I have a question. I wonder what are the advantages and disadvantages of synthetic life compared to natural biological system about application fields in modern biotechnology or other applicable biological sciences?
Yeah, sure. So the advantages of synthetic life, sorry, in general, if you were to synthesize life, right? Yes. I think this can be applied in a wide range of applications. And so we're thinking of evolution being a super powerful tool. And we're already using this in directed evolution, right? So directed evolution is this idea that you use E. coli or bacteria to produce enzymes that are more effective to solve certain problems. Now, if you would do that with synthetic cells based on the central dogma, so based on the biological molecules or the molecules that we have in life, you can expect enzymes, you can expect catalysts that follow biological rules. So they will work in water, they will need water-soluble substrates, they can convert molecules that are only soluble in aqueous environments, for example. So you can expect outcomes that kind of focus on life as we know it. If you were to do exactly the same tricks with synthetic life, so directed evolution of synthetic life, you can go beyond what biological life can do. And you can now start introducing solvents like chloroform, because you can make a life. If we understand how to synthesize life and we're getting good at this, we can synthesize a life form that only operates in chloroform. So if you have a chemical reaction that only operates in chloroform, we can find a synthetic life form that develops or that evolves a catalyst for that. So basically what I'm trying to say is that life as we know it, and this sounds a bit weird, is a little bit limited. It's not at all. And life is amazing, of course, as we know, but It's somewhat limited to a certain set of conditions. And a certain set of conditions is, of course, life is very diverse, but it needs oxygen, it needs water, et cetera, et cetera. If you now synthesize synthetic life, then you can do more than what actual biological life can do, because you can expand those conditions.
Thank you very much.
And also, I have a question too. I'm curious more about conservate-based part, and I'll ask a question from there. If we create a hydrophobic barrier, such as hybrid protocells ones like vesicles, can we take advantage from time about the exchange, the RNA molecule between protocell systems? And also, I would like to ask, has the vocal formation that you said before got benefits like in modern cells ones?
So the first question is, how about like hybrid forms, right? So can we combine protocells based on vesicles with coacervates? Yeah, that's a good point. So the benefit of coacervates is also their downside. It's actually something that Karsten, who's on the slide right now, I'm not sure if I'm still sharing my screen. Yeah, Karsten said it very nicely. Their benefit is also their downside. And the benefit is that they just take up stuff from the environment, right? So that's why I absolutely like working with them. You give them a replicator, it goes inside. Yeah? Or you give them building blocks for the replicator, it goes inside. But that's immediately their downside. They also lose their genotype, they lose their RNA and DNA molecules all the time. And so, absolutely good point. What if we then give them some sort of a cell wall, a synthetic cell? And these are indeed things that we're thinking of right now. It's like, can we have a replicator that produces the building blocks in order to build a cell wall so you don't lose your stuff all the time? Yeah. But you do need to think, so that would also prevent fusion, for example, which is another major downside of these droplets that fuse. So yeah, if Jimmy fuses with Timmy, what is left of Jimmy, right? Vesicles are helpful there, but I find a major downside remains for vesicles. If you work with them, you need to find a way to bring in hydrophilic molecules. And so since it's an aqueous compartment, most of the chemistry in a vesicle happens in water. How do you get RNA into a vesicle, or how do you get DNA into a vesicle? That's very difficult. So then you need to think about how do we make pores and these kind of things. And there are all kinds of cool chemistries for that. But kind of coacervates solve that problem. So you ask me, I think coacervates are more likely candidates at the origin of life if we believe protocells first. And then indeed, as you're saying, maybe some protocell figured out how to make a cell wall, and then from there on, it all started. But I don't know. I don't think we'll ever find the answer anyways. But I think synthetic life based on coacervates may be a little bit easier to think of this transport problem.
Yeah, I think like that too, but the vocal formation question.
Yeah, what I didn't get your second question. Could you repeat that? Sorry.
Yes, of course. Has the vocal formation got benefits like in modern cells ones?
Local formation of biomolecular condensates, or what do you mean?
Yes, biomolecular condensates.
What do you mean with local formation? The fact that it's being produced in the condensate, or what? You
said that the coacervates formation creates a vocalization.
Oh, vacuole. Sorry. Yeah. Sorry. Yeah.
I meant that.
No worries.
And in modern cells, we have vocal forms like.
I don't know. Yes.
Has a some specific detail on it, and I'm curious about that. Has this vocal formation got benefits like in modern cells?
I don't know. So I do not know if it's the same mechanism. We see it everywhere. So when droplets dissolve, we basically said, well, there's stuff coming from the outside and stuff leaving, and the stuff that is leaving is faster than stuff coming from the outside. So naturally, you're going to grow a shell. And this mechanism is true. We also proved that in another paper. But there is other ways of forming vacuoles. If you take a droplet solution and just add a ton of salt so that it rapidly dissolves, or if you change the temperature, then this is a dissolution process. The core dissolves first. There is surfactant-like molecules that are in biological context. So there's proteins that have amphiphilic properties. They tend to help this vacuole formation. So we see this in biology all the time as well. And yes, it has certain benefits. It's also, you could put another droplet inside. So this is a different environment than the outside. Unlike a vesicle, the concentration of molecules here is different than there. And that's maybe what cells also use. So I don't think we understand I definitely don't understand biomolecular condensates, but I think the field doesn't understand biomolecular condensates in biological context sufficiently yet. I mean, every month there is a new paper that discovers a new biomolecular condensate that does something really, really cool. So it's a cool time to be alive. And I think there's plenty of things that biology does with them, and there's a lot to discover, but I'm not the right person to answer that question.
Okay, thank you so much.
Değerli konuklarımız, bir sonraki oturum için 10 dakika ara verilmiştir. Üçüncü oturumumuz saat 12:25'te başlayacaktır.
Esteemed guests, a 10-minute break precedes the third session. The fourth session will begin at 12 p.m. And additionally, you can participate in our giveaway that will take place at the end of the day by filling out the form shared in the chat. To confirm your attendance, you can access the participation form via links shared in the chat and during breaks.
Değerli misafirlerimiz, şimdi Sayın Profesör Philip Engi'yi ilkel RNA'ları haritalamak için makine öğrenimini kullanma konulu sunumunu yapmak üzere davet ediyoruz.
May I now invite Professor Philip Ney to present his talk on using machine learning to map primordial RNAs.
Hello, everyone. Thank you for the organizers for the invitation, and thank you for following this talk. So today, I'm going to talk about how we could diversify autocatalytic RNAs. This work has been done mostly by Camille Lambert, who was a PhD student who did the experiments, and Vaitea Opu, who was a postdoc who did the models. And this work was done at ESPCI Paris. This is the engineering school where I work. It's a school of physics and chemistry that is located at University PSL. And this work is also a collaboration with Mateusz Merlak from the Max Planck Institute for Mathematics at the time, Martin Veit, Francesco Zamponi from Sorbonne University, and Eric Hayden from Boise State University. And also Francesco Calvanese did the models, half of the models. Yeah. So this is a club for origin of life. So I guess you're all very familiar with the origin of life question. Still, it's good to think a little bit about what is the question, because there are many ways to ask this question. What I want to highlight here mostly is that it's a specific question, as we all know. So we are wondering about the process of abiogenesis, which is how you transition.
Sorry for that, but we can see your slides then when you change it.
You cannot?
Yes. Yes, thank you.
Yeah, but then I should pull it full. Yeah, then there is a problem with
We can continue in here like that.
Yes.
Full
okay,
screen.
if it's fine with you.
Yes, it's fine. Thank you.
Okay. It's even easier for me. So. Yeah. So how do you go from abiotic environments to dividing cells? And of course, this is a very process with many steps. And as a scientific question, what comes to mind first is a historical question. We are talking about what happened in the past of Earth, where with an uncertain context. So this is typically something hard to study because we are used to observe something and try to explain it. The origin of life also is not observed re-happening. So even independently of the historical question, it's very hard to study something that you cannot observe. And that was not clear before we knew about microorganisms, and it's Louis Pasteur who demonstrated that there was not a spontaneous generation of life all the time, but it was due to the presence of microorganisms which grew, and also some microscopic forms of life that we didn't see at first and then seemed to emerge from something that was apparently non-living. And the other problem is that you cannot do a direct backward inference of the origin of life from life as we know it. You can study the tree of life, and that may lead you to Yucca, but going very much below that, that's really hard. So, so of course we restrict the question and wonder, okay, is there a way we could focus more on the chemistry? So the famous Miller-Urey experiment shows that you can make a lot of the building blocks of life from simple chemicals. In fact, water, methane, ammonia, and hydrogen. And then you might try to go to, the video doesn't work, but here you would see a video of DNA replication where some molecules are being copied, and still the gap is absolutely huge between the two. So we try to develop scenarios of origin of life and see how there could be a form of self-organization in chemicals during abiogenesis, so this transition. Trying to decompose it in several stages, and what I will show you is about one particular type of object at one particular stage. So the general scenario we try to study in the lab is we start from oligomers, so we don't study how the monomers or even the oligomers form, which is a big question in itself. But we try to see, once you have RNA oligomers, how they may self-organize, for example, form self-assemblies. And then you might have a catalytic feedback, meaning that some of the RNAs will help catalyzing the formation of other due to the action of ribozymes. So ribozymes are catalytic RNAs. And that's what I'm going to talk about today. And other things we do, but I will not talk about, are how those RNAs could start to copy other RNAs and amplify somehow information. And this is where my group stops. Basically, other groups like the Phil Holliger group, for example, or Joyce group are working on building replicases, so RNA polymerase that copy their own template. And at some point, as you have seen in the talk before, it's important to think about protocells, compartmentalization more generally. So this is something we also study and that may have played a role very early, as you have seen. But here I will focus on catalytic RNAs and those that may have the property to make copies of themselves in an autocatalytic sense. I will come back to that. So RNA is a very important model for origin of life because, as many of you may know, you have the central dogma of biology where DNA is replicated. RNA, you have messenger RNA, so DNA is transcribed into messenger RNA, and then RNA is translated into proteins, and proteins are seen as the catalyst. And in this story, RNA is considered as an information carrier. So that role is not questioned since it was discovered in the 60s. But it was discovered in the 80s that RNA can be catalytic. So you can have ribozymes, as I told you. And tetrahymena was the first ribozyme that was discovered. It's a golden molecule here. And so this kind of changes the game because you can imagine that RNA may have played some roles that we attribute to proteins because it can be catalytic. And what often happens is that the RNA catalyzes reactions between RNAs. And that's what was discovered first, and that's what we will use. Well, so the RNA world hypothesis is, of course, a hypothesis, but it has only been reinforced by the discoveries in biology and molecular biology so far. So you find the catalytic RNAs playing important roles in cells like telomerases, spliceosome, but most important one probably is the ribosome. So the ribosome that does the protein translation is made of RNA and proteins. But it was understood in the years 2000, thanks to crystallographic structures, that this RNA, this ribosome, it is the RNA that does the catalysis. So the ribosome is in fact a ribozyme. So if you imagine now what's happening with translation, you have an RNA, the ribosome, that's taking a messenger RNA and transfer RNA, and all this RNA machinery is making the proteins. So this observation, this discovery points further to the RNA hypothesis as a valid hypothesis. So once you've said that, how could life have started with RNA? And so this is why several groups work on trying to find an RNA self-replicase. So it would be an RNA molecule that folds and acquires the catalytic properties of being a polymerase, and a polymerase that catalyzes a copy of RNA. And if in particular, this polymerase copies its own sequence, or a copy of its own sequence, then you could have the start of an evolutionary process that is similar to what we see in biology, where you have heredity by copying, you have mutations causing variation, and the speed of copying, for example, could lead to a form of selection, of competitive selection. And you would have all the properties of Darwinian evolution. So this is here a paper of this was a summary of how long an RNA could be copied by another RNA as a fraction of its length. And in 2013, there was this article from the Oliger group showing that you can indeed copy an RNA. There are RNAs that copy RNAs as long as themselves, but they could not copy themselves at the time. Now it's maybe changing because there has been a publication from the Joyce group where an RNA copies not itself, but other catalytic RNAs that are smaller from nucleotides. And there is also a preprint from the Oliger group indicating that you can copy an RNA. An RNA can copy itself, but based on trimers and hexamers. So this is a big progress. I don't know when it will be published, but it's a preprint. And then, the next step would be to go to replication with nucleotides. So at the moment, there is not an RNA self-replicase in the canonical sense that can copy itself from nucleotide, but there are objects that start to get there. That being said, those objects are very complex, and as you imagine how it could have started, you have to find this object, and it has to meet its exact copy by chance. And also having this object appearing in the first place, a long object is hard to imagine. So what we are studying is what could have been more rudimentary stages of evolution, where more rudimentary catalysts could have helped the appearance of more complex catalysts. In this regard, there are at least two experimental systems that are well known and that amplify autocatalytically in the laboratory. So there is this, I will call it Azoarchus because it's a ribozyme, an RNA that comes from the Azoarchus bacterium. It's a self-splicing RNA, which means that it extracts itself. It's an RNA that extracts itself from a messenger RNA. So it folds, and you see the fold here, and then it cuts RNA bonds and makes RNA bonds. And so the Lehman group at the time has used this property to have autocatalytic self-amplification. So if you cut it into pieces, which are colored here and called WXYZ, those pieces can self-assemble. I'll show you a video. And the final covalent object can make the covalent bonds between the pieces so that you have a second copy. And there is also the Joyce reciprocal ligase that you see here. So here you see one step of ligation where the bottom part is attaching two fragments on the top. And once those two fragments are attached, you can have the reciprocal catalysis. So you can also have amplification. So you see those two types of objects are similar in their functioning, but overall functioning. So it's autocatalytic. You don't have an external template, unlike in the self-replicase. But they are also a little bit different because the degree of base pairing between the catalyst and the substrate is very large in the Joyce system, whereas it's very small in the Lehman system. So we will use in the talk the Azoarchus here on the left, and here is a video that facilitates the understanding. So you have RNAs, RNA fragments, oligomers, and we don't discuss how they form, but this is a model, so they are a little bit big at the moment to be really realistic. But by base pair complementarity, those fragments can find each other and form a complex. And this complex is non-covalent. You see that you have some ends that are not attached. But if you have a covalent version of this object that arrives here in purple, this covalent version is going to attach the non-covalent parts to make a stable molecule. And so you see you have a form of photocatalysis where you start with 1, and then you have 2, and from 2 you can make 4, and so on. So you can have amplification, but using rather large oligomers to start with. Objects, both this one and the Joyce reciprocal ligase, have been diversified a little bit in the substrate recognition region. But we don't know if this form of RNA autocatalysis is something relatively banal or maybe something completely exceptional that has been engineered in the lab, because mostly we know two catalysts that can do that. So this is a question we start to address with the work I'm going to show. And we were wondering, can we leverage evolutionary information and RNA folding predictions to show that there are potentially many of those autocatalytic RNAs? So to give you some feedback on the type of methods that exist to diversify RNA, the most famous one is in vitro evolution, where you take DNA, you make copies with mutation, with error-prone PCR, and then you do transcription, and you have a lot of RNA molecules with mutations that would be in red here. And this is how the RNA replicase, for example, is being studied. Like, people are trying to do directed evolution to find such an RNA replicase. So this is artificial evolution, where the DNA copying and the mutations and the selection are all done artificially. But this is a way to explore the sequence space, because you can start with very, very large libraries of random RNAs, like 10 to the 15 different RNAs. So this is extremely big. There are other studies that took very, very small RNA and tried to exhaustively make all the mutants to look for an activity. And this is still relatively recent work. They found that you had kind of isolated islands, so isolated peaks of activity, which were very loosely connected by point-wise mutations between them. And you can also do what's called deep mutational scanning. So it's very similar to what was in the middle. It's like you really make every possible combination of every mutation at each position and combination of mutations generally, because if you do more, it's too big. And instead of selecting the best one, like you do in directed evolution, you really measure the properties of every mutant you have made. And you see here the matrix will be kind of a representation of all covariations that matter. So when you mutate two nucleotides, so it's a sequence versus a sequence, when you have a dot, it means here combining variations in those two positions at the same time is very important. Or when it's light colored, it's not important. So this is a way to study the constraints in the molecule. And where you see a lot of reds, generally it corresponds to a region where you have base pairing. But not just that. And we hear about artificial intelligence every day now. So there is a lot of hope in doing computationally guided generation, design of biomolecules. That started in RNA, I will show you, with trying to use secondary structure prediction. So this is not artificial intelligence, but it's really computing how the RNA folds and trying to see if we can design new RNA using those tools. And with machine learning, trying to do several stages now, making mutants. So Hamming distance here on the y-axis means the number of mutations you have made, and then you have a score, so you measure experimentally. And from this stage, you can see, okay, those mutations are good, those are not good, or I must do those together. Keep them and do a next round of diversification and try to learn the rules of diversification of particular objects. And, well, you've very certainly heard about the Nobel Prize of Chemistry this year, which was for Google DeepMind AlphaFold. And AlphaFold is a large language model, so it's machine learning that is going to learn across all proteins the type of variations that occur together. And the power of this method is to learn under what context, so the context of other mutations, do some variation occur. It is the same that when you talk, you use some words and then the word that you use means something. It's going to be different whether you use other words in the sentence or not. And the same principles apply to proteins, work well. Of course, there are additional features, like introducing some physical constraints in the design. And that's such a success that only a few years after the first publication of this model, the Nobel Prize was attributed. So this gives a lot of hope that you can design RNA. But AlphaFold works very well for proteins, natural proteins, but it works very badly for RNA for the simple reason that there is not much database. The database is too small. The other point is that AlphaFold predicts the fold, as its name indicates, but it doesn't predict the function. So it's not immediately clear how you can make a catalyst from AlphaFold. Of course, it can be used to design catalysts because the fold is very important for catalysis, but it's not direct. So there is another method that was applied by colleagues in 2020, which is simpler somehow algorithmically than AlphaFold, but that looks at evolutionary covariation. So you look at two positions in the, how correlated are positions within a protein, a family of enzymes that do the same thing. Here it was the chorismate mutase enzyme. And you use this statistical information, I will show you how, to design new ones. And it was shown that it's quite successful. So because for RNA, we cannot use AlphaFold, and it was not available when we started the project. But we knew of this work, and our colleague Martin Veit from Sorbonne University developed this method. That's what we decided to use. So I don't want, this is the only equation, so I don't want to annoy you with that, but I can tell you the principle. So it's called direct coupling analysis. And this name means that if you imagine a biomolecule that folds, it can be very cooperative. So you have contacts, but you can have interactions between three parts, or it can be very complex. And here the idea is to learn a statistical model that tells you pairwise, so what is the degree of influence a position has on another position. So concretely, if you take a RNA here that's folded, and there are quite stereotypical positions that have been indicated in green. So in green, you have a position that's just here to make sure that it's long enough. In red, you have two positions that are in base pairs, so Western-Crick base pairs that are in contact. And in blue, you would have an imaginary active site for the enzymatic activity, so catalytic activity. If you look at this same realization of a ribozyme, in our case, RNA, across different organisms, you will see that the green residue, so every line is a different organism, so you look at the same function in different organisms. If it's just there to ensure that there is a good length, it doesn't matter which letter of the RNA you have. So this is going to look very variable. There is no conservation, basically. If you look at the active sites, maybe you absolutely need a G. This is the case in the Azoarchus ribozyme. Then this is going to be highly conserved. And you will see statistically a very big difference between green-like residues and blue-like residues. But for contacts, it's a little bit more complicated because you know Watson-Crick base pairing. If you have an A, to have a contact, you need a U in front of it. And then if there is a mutation to a C, you cannot keep the U, you have to change it to a G, etc., etc. So what you have here is not just conserved or non-conserved, but it's co-evolving residues. They are not conserved, but they evolve in a coupled manner. And this is what this model is trying to learn. is trying to learn the position-wise frequencies and the coupling between positions that reproduce the best the distribution of nucleotides that we see in natural sequences. So then you need natural sequences. And the Azoarchus ribozyme is one particular example of what's called group 1 introns. So group 1 introns are those RNA within mRNA that fold and splice themselves out. And there are many of them that are available in, I would say, in databases in the order of 10,000. And this is how they look. So they are all have a general structure that's relatively the same, but there are variations in the composition of the bases, of course, but also the presence of the loops here, for example. So we extracted those that look like the Azoarchus ribozyme to have a consistent data set to learn from, and this data set contains approximately 800 sequences. You see here the RNA, how it looks in 3D. So you have this kind of helices that cross each other. And in the middle, where you see the yellow dots, this is where the catalysis is happening. So here is a representation in the middle of the same structure, but spread for the sake of representation in 2D, where you see that certain regions are base paired, you have some bulges, you have some loops, you have some tertiary interactions. So this is a group 1 intron family, and we extracted more particularly the Azoarchus sequence. And here you see the tree, kind of the tree of life, the phylogenetic tree, where Azoarchus, our model, is one particular dot among all possible variations. And here you see a long, so this is now a 1D representation of the sequence, and this is called, so this is a consensus sequence, where at each position, if you have a color, it means that you have a certain letter at a certain frequency. So you see that there are a lot of variations, a lot of different implementation of this molecule in nature that use different letters, and some regions are very highly diversified, some are more conserved. So what we did is that this mathematical model I showed you, we fitted to these 800 sequences to find the coefficients that reproduce the best the natural diversity. And we could see before doing a further experiment that this was relevant, simply because if you look at former data from other colleagues, so from Eric Hayden, from Boise State University here, where he has generated tens of thousands of point mutants, so a few mutations, you can, if you use this score, so this is the score of the model, you can distinguish the mutants that resist to different concentrations of magnesium. And so here the peak of the distribution corresponds to selection in different more or less aggressive environments. And you can see that the mathematical score is telling you, yes, this family, indeed, those mutants are better or worse. The other interesting aspect is that the mathematical model indicates relatively good predictions of the contact. So this is called the contact map, where you have the sequence on the x-axis and the same sequence on the y-axis. As I explained you before with one of the examples, if you have a dot here, it means that there is a contact between this position, which would be 110, for example, and this position, which will be position 75. When you see a line like here, a diagonal, it's what you find when you have a helix, because you have a succession of positions that are in contact with each other in reverse order. So below is the true contact measured with the crystal structure, and above is the prediction of the contact that has never seen the crystal structure, but has just learned the most probable model from the observed diversity. So it's quite striking that it's not exact, but you find a lot of the existing contacts just by looking at the letters. Additionally, in RNA, colleagues from Vienna, and there are other packages, but this is the most famous one, and this is the one we used. It's called Vienna RNA. Without knowing anything about natural diversity, you can take a sequence and compute what is the most probable fold and establish a contact map like I showed you before. So this tool is good for small RNAs. It works very reliably up to 100 nucleotide long RNA, but above that, I mean, you can use it for larger RNAs. More than 150, it starts to be problematic. And our RNA is 200 long. So we are not necessarily in the good region, but what we did is that we still used it because we think it's going to constrain the structure that can be made when we generate a new sequence. So indeed, we see that the prediction is not perfect, but it catches some parts of the molecule. And so that's already informative. So here, this is very technical, but you have several algorithms, UPAC or RNA fold. And just to say, we use RNA fold, which does recover some contacts, but not what's called the tertiary interactions and the pseudonodes for those who are into RNA. So you have regions where the molecule is folded in a more complicated manner, and that the algorithms cannot catch. Again, we use the same benchmark of this older data set with mutants, and we find that we can also recover some score from this other model. So we have two models. One is learning from evolutionary data, and one is predicting ab initio from physical mechanisms. And so that's what we thought, okay, we should try to use that. So we developed a workflow where you use, on the one hand, the sequences found in nature. So you see here this colored rectangle. Every row is a different organism. So there are group 1 introns in many organisms, bacteria, plants, humans, shrimps, dogs, cats. You look at them. You do what you call an alignment of the sequence. So you try to put this sequence as well as possible aligned. So which nucleotide is supposed to correspond to which nucleotide. And you see that you have some noise. So this corresponds to the base variation. Because those sequences have different lengths, I don't explain you, but we had to crop them and to make sure they are perfectly aligned, not with deletions or insertions. In parallel, we took the Azoarcus sequence and we can run the folding. So we take a candidate sequence, not Azoarcus, but Azoarcus with mutation that we generate in the computer. And we can try to predict the folding structures. So we have the DCA, the statistical model information, and the folding model. And we combine those two in a score. And at this stage, it's a very simple combination. We just add, it's a weighted average of the two scores. So more or less, we just add the scores. And we say, okay, this new sequence is probably good if it's both good, similar statistically to natural data, and also respecting to some degree the prediction of the structure that we know. This was for the modeling part, and what's very nice in that system, we can do high throughput experiments. The experiments, we don't screen for autocatalysis, but we screen for something that's simpler to screen, which is the natural activity of those catalytic RNAs. So here, the candidate RNA is represented in black, and it's synthesized with a sequence on its 3' end, so one extremity of the RNA, which is A that you see here in green. In the mixture of the experiment, we put a substrate in red that's called sub1 here, and the RNA has to pass a test. And the test is to attach the green part A to the substrate 1, to exchange the substrate 1 for substrate 2, which has a B extremity, and to attach the orange part to itself. This is mimicking what's happening in nature. And what is very interesting about this assay is that by sequencing the molecules, you will see those that still have an A on their extremity, and those that have sub2, so the orange part on their extremity. And so all the mutants that you are going to look at, you can say, oh, like 10% of the time it managed to attach the orange part, and 90% of the time it didn't. So it's active, but not very much. Or one that would attach 90% of the time the orange part would be a very good catalyst and would have a high score. The next important thing is that those type of experiments, we can do them at a high throughput, which means that we can order DNA from a company, a DNA that comes out of the computer. So we generate, for example, 12,000 different DNA sequences, which are here of length 200 or 250 to have the full construct. The company delivers us exactly the sequence we want, but in a pool, so they are all mixed. But the assay I just showed you, it works in a pool. So we add the substrates, and then by sequencing, we recognize the different RNAs we have designed by computer, and we also measure how often they have attached the orange part to themselves. And this gives us, for each sequence, an activity score. So the experiment itself is very short. You just mix stuff, you incubate 1 hour, and after you have to prepare your library for sequencing. So you take all your RNAs that have more or less reacted, you convert them into DNA, and you send it to the DNA sequencer. And after, you have to do the bioinformatic analysis. So in this way, we can score very rapidly tens of thousands of RNA. And that's what we did. So we assayed more than 50,000 sequence. This particular data is based on more than approximately 24,000 sequences that have been assayed experimentally. And the game we played is to compare many different models. So this is the most important figure of the talk. On the x-axis here, you see the number of mutations we introduced in the RNA. So 0 is our reference point, is the Azorchus molecule. So we know this one is active because this one was published. And here you see we introduce up to, let's say, 100 mutations. And the different colors correspond to the average score, so the fraction of molecules that are successful in each bin of 10 mutations. So if you do random mutations, so let's only look at the models on the bottom panels, and here the bottom left. If you introduce random mutation in a molecule, you lose the activity very rapidly. So after 10 mutations, the curve goes very close to 0. So this means that even though the molecule is 200 nucleotide long, it's very easy to kill the activity. After just a few mutations, if you do it blindly, you are going to completely lose the activity. And now we compared different models that either use a structure prediction or use a statistical information or a combination of both. And I'm not going to detail all those models because it would be quite lengthy. But you see that there are differences in their ability to introduce mutations. Some of them are very poor, like the random mutations. Some of them are intermediate, so you see the red or yellowish curve. And here is another range of models. Some are better and better, and one is particularly good, is the purple one. So all the curves that you see on the bottom left panel are those generated with structure prediction. So we can see that using structure, we can introduce more mutations than with random. You see up to 30 mutations, I would say, and still make functional molecules. So this is already quite a number of mutations, but you cannot do very much more. Whereas if you use evolutionary information, so what nature has explored, and you fit the statistical model, that's DCA, those are the green, blue models. You can introduce much more mutations. So you can make new functional objects with up to 60 mutations. And the purple one is very interesting because it is where we combined the physical models and the evolutionary statistical models. So you see that by combining all sources of information, this is the maximum you can do, basically, the maximum information you have. You can exploit it, and you can make mutants that are very successful mutants with many mutations, so up to 60 and even 65. I'll skip that. So one first question would be, okay, we can introduce mutation, but are there some parts of the molecule that are hard to diversify or not? And here you see the molecule, it's a heat map. So the red parts are those that we can mutate very easily. So we can make artificial molecules by varying those regions, but the green part are hard to mutate. And you see kind of a diagonal here. And it's very logical because this region is, when you look at the 3D structure, it is a catalytic core. So this must be very well organized. It's a very entangled region. And nature has not diversified it very much. And we could not diversify it very much either. So there is a challenge here to expand even more the diversity of molecules, which be to diversify the catalytic pocket. The other point I could highlight, here you see a 1D representation of the molecule, so from 0 to 200 nucleotides, and the frequency of the bases of the different models. And what we see is that by adding the structure prediction, so the physical model, you can introduce a mutation in purple. And if you compare it to blue, which is just the evolutionary signal, it shows that we could introduce diversity that's not present in nature. That's very interesting because it points to the fact that we could go beyond natural diversity and not just interpolate. That is the criticism for machine learning in general. It can learn what exists, it can interpolate between what exists, but it cannot really extrapolate. It cannot create novel objects. But if you combine statistical information with physical models, we see here that you can start to generate completely artificial objects that, and you extrapolate a little bit. And indeed, we see that we can manage to go further in diversity. To close the loop, the assay I showed you is a mimic of the original activity of the RNA in nature. But this activity has been used to engineer autocatalytic RNAs. And so our reasoning was that if we find mimics of the natural activity, then we can reapply the same engineering skill to have autocatalytic RNAs. And so that's what we tested on random mutants. We took 20 of them, so I show you the result for 8 here. We cut them into parts, we incubate them separately, and we see that, if you're used to electrophoresis, those are different time points. So for example, for candidate 5, this is an incubation of smaller fragments. And you see the appearance of a band, which is a larger band because it's upper. And this larger band indicates that you have formed the full-length covalent product from only fragments. So this proves that there is an autocatalytic process going on. So more than 60% of the sequences that we tried were indeed autocatalytic. Sorry, I'm going to sneeze. No, it's okay. Sorry. So this indicates that our proxy, our high throughput proxy is good. It's at least 60% of the time, or let's say more than 50% of the time, it is predicting autocatalytic activity. So where are we in terms of number? We tested 24,000 RNAs. We found of the order of 10,000 that are active. So this is more, this is as much as is known in nature. So artificially, you could design new objects and design as many objects as are already known in nature. And by our sampling, we know that at least half of them are autocatalytic. So now we go from one known sequence or two to 10,000. But this is just a little glimpse about the space, because experimentally we could only test tens of thousands, which is already a lot, but in reality the number of possible objects is much larger. And so my colleagues found a good trick to calculate the number of objects that we could have made. And here you see those curves. And I think those are really very novel type of curves. On the x-axis, you have the number of mutations that have been introduced in Azoarchus. And on the y-axis, you see the number of sequences that you could generate given the model. In blue, you have the curve, which is the total number of sequences that could be generated by putting random mutations. For example, if you put 20 mutations, you could have 10 to the 33, say, different sequences. And of course, doing random mutation is the best in the sense that you allow yourself to do whatever. So this is the curve that is on top. It's a blue curve. But there is a problem because, as we saw, if you do random mutation, at some point, you lose the activity. So the random model tells you that you have less than 1% activity after 14 mutation, and it corresponds to this blue star, which means that in practice, combining the model and the experiment, you can only assess that there exists. This is where your experiment stops. So you can only say, okay, from random generation, I can say that there are 10 to the 28 different RNAs with this catalytic activity. And then you could look at better models in terms of their ability to introduce more mutations, but they are more restricted because they impose constraints on the generation. So the curves are below. And to give just the final result, so you can compare those models, both combining how far they go on the x-axis, how many mutations you can successfully introduce, but also how far they go on the y-axis, how many diverse molecules they can make. And the best model was the DCA, indeed. Not the DCA with structure, in fact, but the DCA, the simple statistical model, tells you that there are 10 to the 40, approximately, sequences that could have this catalytic activity. So this is an estimate of the number of autocatalytic RNAs that have this specific structure that you have in the space. And 10 to the 40 is an absolutely huge number compared to 1 or 2, which was the state of the art at the beginning. I could stop here. I don't know what you think, the organizers.
Since we started late, you can continue if you'd like to.
Okay, I'm going to show you this last slide.
All right.
Which is, okay, is what I said general somehow? Is it very specific to this RNA family? And it was very striking to us that it's potentially very general. So we looked at other families. All the families of RNA we could find between lengths 50 and 250. And we did the same estimate, like how many sequences can you generate with the different models? The blue, this is now a log-log curve. So this is why you have straight line and not lines that are curved like before. The blue curve is a random model. So it's all possible sequences you could have of length, say 100. And so this is, here it's exponential to the 200. The black curve is the number of sequences that you get if you impose the secondary structure with a physical model. And so you see it's a curve that is below, but strikingly it's a line that was not obvious. So you have a scaling law. And you could say the number of sequences I can make of a certain length with a given structure is 4, so the number of bases, to a certain power. So there is an effective diversity that you can use. So if you can do any nucleotide, it's 4 letters plus a gap. So the effective diversity is 5. So the number of available sequence is 4. It's 4 to the 5 to the length of the RNA. But if you impose secondary structures, the effective diversity varies like a power law, so it's an exponent, and the effective number of nucleotides that are available is a little bit below 3. If you use conservation of single positions, so the statistics of letters of single position, it's approximately 3 also. And then if you use a coevolution signal, which is the one we used to generate successful objects, you find that it's approximately 1.8. So this tells you what? This tells you, okay, I have a certain function, self-splicing in our case, or amino acylation, something else. And I know that in nature, there is a family of lengths, say 100, that achieves this function. Then I can say, I can compute from this exponent the number of sequences potentially in the whole sequence space that could do this activity. And so the project I showed you before is a specific point here, but this shows that there is a general law that spreads at least between 50 nucleotides and 250 nucleotides, which is the range of lengths of interest for origin of life. Okay, so I'm going to stop here. So the conclusion is that we could populate the sequence space with new autocatalytic RNA. So we self-reproducing in the sense of autocatalytic, not in the sense of template-based replication, using an evolutionary model coupled with a physical model that predicts secondary structure. And we could use high-throughput experimental screening to assess the success of those different models. And so in practice, we identified, actually now it's more than hundreds, like thousands of active ribozymes with up to 60 mutations. And we also found that the artificial ribozymes typically are also self-reproducer. And finally, we could estimate a number of self-reproducing autocatalytic RNAs, which is of the order of 10 to the 40 in the space of sequences of length 200. So I'm going to stop here. Thank you.
All right.
Sayın Profesör Philip Engi'ye konuşması için teşekkür ediyoruz. Kendileri adına Tema Vakfı'nda filan bağışında bulunduk. Bağış sertifikasını e-mail üzerinden ileteceğiz. Şimdi 5 dakikalık soru bölümüne geçiyoruz. Katılımcılarımızı sorularını sormaya davet ederiz.
We extend our sincere gratitude to Professor Philip Nigi for his presentation. We made a donation of saplings to the Turkish Foundation of Combating Erosion, Reforestation, and the Protection of Natural Habitats on their behalf. And we will send a certificate via email. We now have 5 minutes for questions, and we encourage our participants to ask any they may have.
We have a question from the chat. If different RNA sequences are formed in a prebiotic environment, what kind of a selective advantage could those with autocatalytic activity have?
Yes, so it's a very good question. I didn't comment very much on that. The general idea is that if you have an autocatalytic process that uses smaller building blocks to build larger molecules, you could have an enrichment of those larger molecules due to self-amplification. And this enrichment would be not a form of evolution, but maybe the first form, the beginning, the starting point, where you enrich the mixture of RNA in catalytic RNAs that could further process other RNAs. And it's really not obvious that this could occur. So behind this question, there is a problem. I show you sequences, they can self-amplify in a mixture where we only put the fragments. But what happens in a random mixture in the context of origin of life? That is really not obvious. And maybe those autocatalysts, they can exist, but they don't work because there are many parasitic interactions with other random sequences that are around. So this is a big problem to solve in the context of origin of life. Yet there is hope because some colleagues in Japan, Ryo Mizuchi and Ishihashi, they have mixed random RNAs of length 20, I think, or 10, I don't remember, activated RNAs, and they found the emergence of a specific RNA in this random soup that seems to amplify by autocatalysis. So it seems to be possible, but this is where we are. It's very recent, and we have to show that such process can go on and maybe lead to complexification.
And I have another question. The RNA world hypothesis proposes that autocatalytic RNAs could evolve through natural selection. However, how likely it is that these molecules with autocatalytic activity will form among the randomly synthesized RNA sequences?
Yeah, so I answered a little bit to this question already in my other answer. There are recent experiments showing that you can seemingly have autocatalytic RNAs coming from those random soups. So in this first step of the project, the first question is, do you have many? So is there a probability that there exist autocatalytic RNAs? But as I told you just before, this is only one part of the question. The other part of the question is, could they survive? And to survive, they should have very specific interactions with their complementary RNA. So I think this is one part of the answer, and we don't know. We don't know the answer for the moment. We have to think about which RNAs are able to more favorably recruit the good other RNAs. And the second part of the answer would be to design catalytic RNAs which are as small as possible. So this is what we plan to do next, to try to do the same as we did, but minimizing the size of the catalytic RNA with machine learning also.
Right. Thank you for your answers and thank you for your time. I think that's all. Yes. So can we just take a break from
Thank you very much. Yes.
Ben
Zeynep.
Şeyinde miyiz? Ben kapanış yapayım.
Tamam.
Değerli katılımcılarımız, Astrobiyoloji ve Yaşamın Kökenleri konferansında ilk günün yarısını tamamlamış bulunmaktayız. Yarım saatlik 40 dakikalık aradan sonra kaldığımız yerden devam edeceğiz.
Esteemed participants, we have now completed the first half of the first day at the Astrobiology and Origins of Life conference. Now it is time for a lunch break. At 2 p.m., we will continue from where we left off.
Değerli misafirlerimiz, aramız sona ermiştir. Şimdi 4. oturumumuz için Sayın Profesör Ian Crawford'u dünyanın ayını keşfetmenin astrobiyolojik nedenleri konu sunumunu yapmak üzere davet ediyoruz.
Dear guests, our break has ended. Now, for our fourth session, we invite Professor Ian Crawford to deliver his presentation on astrobiological reasons for exploring Earth's moon.
Okay, well, thank you, everybody. Good afternoon. It's a real pleasure to be here to talk to your group in this really tremendously exciting field of astrobiology and the origin of life. So, obviously, I've got this picture up. You recognize the Earth in this picture, and the Earth is the only planet in the whole universe that we actually know life has evolved on. And of course, we really want to know whether life has evolved on any other planets. And that really is what the topic of astrobiology is about. But today I was going to talk about the moon. So this picture, it's quite a nice picture. It shows the Earth and the moon together. It was taken by NASA's Galileo spacecraft on its way to Jupiter in the mid-1990s, looking back at the Earth-Moon system. And so here's the moon, and the moon is sort of dull and brown, and it doesn't have any water, and it doesn't have any life. And as far as we know, it can't have any life because it's got no water or no atmosphere, and also a very harsh radiation environment, of course. But despite all of that, I'm going to argue that the moon still has quite a lot to tell us about the history of life on Earth and the possibilities for life elsewhere. And I think the key insight that I'd like to try and get across is that for the last 4,500 million years, the moon has been orbiting the Earth, which means for the last 4,500 million years, the moon has been orbiting the only inhabited planet that we know of in the universe. And it's during this time, things will have happened to the Earth which are relevant for the history and evolution of life. And yet the Earth is a very geologically and meteorologically active planet, and it does have a rich geological record, but only really a rich geological record going back some hundreds of millions of years. The really ancient history of the Earth is largely eroded away, and yet the moon has preserved a record of things going on in the cosmic environment in the Earth-Moon system over billions of years. And I'm going to try and convince you that much of this record is of astrobiological interest. So I've got quite a long list, and I'll just go through it, and then I'll elaborate on some of these points later. A nice picture at the bottom. This is Apollo 17. So this is Jack Schmitt and the Apollo 17 lunar roving vehicle from the last time humans went to the moon in December 1972. But anyway, I'll just draw attention to the bullet points. So what can the moon tell us that's astrobiologically relevant? So the list starts with perhaps the most boring, but actually still really important, I think, a characterization of the impact cratering rate in the inner solar system. So the moon is covered in craters, and it's covered in craters because of meteorites bombarding it over time. But of course, since the moon's been orbiting the Earth, the record of meteorite bombardment on the moon is basically the same as the meteorite bombardment on the Earth. I mean, it would have been a bit worse on the Earth because the Earth is bigger and it has a stronger gravity. But basically, the moon has recorded the impact flux onto the Earth-Moon system, and therefore onto the Earth through time. And this is important because we'd like to know when life originated on the Earth, was it having to survive frequent giant meteorite impacts or not? Well, I think the moon can tell us that. The next point I'm going to talk about is the preservation of cometary volatiles and organic molecules on the moon. The moon's craters at the north and south poles of the moon, which never see the sun and therefore are very cold, probably preserve the delivery of volatile compounds, so water and carbon dioxide, methane, but possibly also organic molecules coming in from the outer solar system. So there's a potential record there of the delivery of organic, volatile and organic materials from the outer solar system to the Earth-Moon system, and therefore by inference to the early Earth, and this may be preserved on the moon as well. There's the possibility that there may be meteorites from the early Earth preserved on the moon. We have meteorites from the moon on the Earth. Probably there are meteorites from the Earth on the moon. And if we could find them on the moon, they could preserve a record of the Earth's environment in its earliest history, which the Earth itself has largely lost. I think the moon will preserve a record of solar and galactic activity, solar wind, solar flares, galactic cosmic rays throughout our solar system history, and much of that record is relevant to the habitability of the Earth. There may be some more exotic possibilities. I've written alien artifacts here, so that may seem a bit strange, but I'll try and justify why I put that on when I get to it. Then there's the more practical current astrobiological or biological research that could be done on the moon, like assessing the survival of microorganisms and organic materials in the lunar environment. And in part, that will be a proxy for, I think the next talk is about Mars, if I remember the program correctly. Mars is also a very important planet for astrobiology, and I could make the argument that we'll learn more about Mars if we send people to Mars. But there's a tremendous amount to learn about the effect of the space environment on biological systems before we could safely send people to Mars. That's we could learn about that, doing that on the moon. So this is my top level bullet list of points. Why I think the moon, although it's dead, is nevertheless of astrobiological significance. So I'm now going to go through these, illustrate some of these points. So this was the most prosaic, the most boring one at the top, characterization of the Earth's impact flux. So the picture here shows you some craters. This is the crater Daedalus. It's on the far side of the moon, but I just show it here to show the moon has lots of craters. And therefore it had lots of meteorite impacts over time. And the flux of these impact craters on the moon, more or less, would have been the same or similar to the flux of meteorite impacts on the Earth. And what's relevant from astrobiology, and I think also from the origin of life, I mean, I realize this is an origin of life meeting. I think what's relevant for the considering the origin of life on Earth is one of the things that's interesting to know is just how many times was the early Earth being hit by giant meteorites? Because really large asteroid collisions with the early Earth could have vaporized the early Earth's oceans or raised them to a very high temperature or sent very serious shock waves through the Earth's early oceans. And these may have been very damaging. You could imagine life just trying to get started somehow, and then suddenly a meteorite impact might have destroyed it, set it all back, and then it would have to try again. So we'd very much like to know what the meteorite impact flux was on the early Earth. But we'd be talking about the meteorite flux on the Earth as it was 4,000 million years ago, something like that. And the Earth, we have hardly any Earth rocks dating back as old as that. But the moon preserves this record. So the big graph here, it shows impact rate relative to the present. It's a schematic graph, of course, but it is logarithmic. So notice there's a huge, for 8 orders of magnitude increase in lunar impact rate sketched on the vertical axis. And then the age going back in gigayears. So if we're talking about the origin of life on Earth, it presumably happened somewhere here. 4 billion years ago, maybe 4.2. I mean, we've got good evidence life was present on Earth by 3.8. So life was originated on the Earth sometime during this time. So the question is, what was the meteorite flux doing at that time? And this diagram shows 2 possibilities. The one possibility is the meteorite rate would have been very high, very, very early on, shortly after the moon formed, 4.5 gigayears ago, then dropped very quickly, then had a quiet patch, and then a sudden spike, which some lunar geologists think they can see in the cratering record on the moon. This is sometimes called the late heavy bombardment, and then going back to a quiescent rate that we have today. Or the rate may not have had this late heavy bombardment spike. It might have declined less slowly from the origin of the moon, but still by 4 billion years ago, it might still have been 1 or 2, a factor of 10 or 100 times its current rate at the time life was evolving on the Earth. And we don't know the difference between these 2 curves, but the moon does know the difference. If we explore enough craters, date enough craters on the moon, we would be able to tell whether there was this late spike in heavy bombardment which would have caused real problems. If life evolved on the Earth, originated on the Earth here, then on the solid purple line, it means it could have arisen in quite a quiet patch with not too many meteorite bombardments, but it would then have had to have survived this big heavy pulse somehow. Or if the dashed line is correct, there was never this heavy pulse and meteorite bombardment and for early life to have to survive through. So we don't know which of these is correct, but we do know the moon has the answer. So we just have to explore the moon, and then eventually we will know what the impact regime was like when life originated on the Earth. So that's the first bullet point. The second bullet point deals with the astrobiological potential of lunar ices. So this is a beautiful picture. This is taken by a Japanese Kaguya spacecraft, probably about 15 years ago now, or 2009, I think, something like that. It shows the south pole of the moon. You will recognize the Earth, obviously, and northern hemisphere bias. Since the south pole of the moon is the right way up, the Earth is upside down. So you recognize this is Australia here. And this will be Southeast Asia over here. But I show this picture because the actual south pole of the moon is there, pretty much where the red laser pointer is. That is the geographical south pole of the moon. And it's close to the rim of this. This is a 20-kilometer diameter crater, and it's called Shackleton. But the point about this image is that there's a lot of shadow. And there's a lot of shadow at the poles of the moon because the sun is always on the horizon at the lunar poles, which means that the floors of craters never see the sun, which means they're very cold, like 40 Kelvin. And under those conditions, even in a vacuum, water ice is stable. So we've got good circumstantial evidence now that many of the permanently shadowed craters, they're called permanently shadowed because they're always shadowed, permanently shadowed craters at the poles of the moon, ice has accumulated. And the question is, where has this ice come from? Where has the water come from to make the ices in permanently shadowed craters? And probably much of it has come from the outer solar system in comets or hydrated meteorites. And we know that comets and hydrated meteorites also contain organic materials, organic molecules, amino acids, or a very wide range of organic materials. And so if comets or meteorites were bringing organic materials to the moon in the early solar system, they'd be preserved here. But they'd also been bringing organic materials and volatiles to the early Earth. So if we want to understand the role that meteorites and comets may have had in bringing prebiotic organic materials and important volatiles like water to the early Earth, then the poles of the moon may preserve this record. So that is something else that's astrobiologically significant. I mean, to the extent to which the outer solar system may have provided volatiles and organics to the early Earth is potentially preserved on the moon, at the poles of the moon, in these permanently shadowed craters. So that was the second bullet point on my list. Then there's the point that I made earlier, that the moon may contain meteorites derived from the early Earth. So here is an artist's drawing of a large asteroid collision with the early Earth. Now, things like this must have happened many times because the moon has many large impact basins, and they date from the time period of about, perhaps 4.3 to 3.8 billion years ago. And we've dated those on the moon fairly accurately now, thanks to rocks from the Apollo missions. So, but if the Earth was being hit by giant meteorites in that time period, then so was the early Earth. And of course, just to go back to my earlier point, this is important from the origin of life field, I think, because the early Earth would have many of these giant bombardments. I mean, this would be a hazard that early life must have somehow survived through this period of giant impact bombardment. But here I'm raising another point. If a giant asteroid were to hit the early Earth like this, then amongst all the debris that gets thrown off would be fragments of the Earth's early crust. And many of these fragments would have ended up into space. and some of those would have landed on the moon. And therefore, we'd very much like to know what the environment of the Earth's surface was like 4 billion years ago, because that is the time period in which life probably arose on Earth. But the Earth itself hasn't retained a record of its crustal rocks from that age because it's eroded them away. Yet a giant meteorite on the Earth 4 billion years ago would have thrown bits of the Earth's crust into space, and some of them would have landed on the moon, where we might collect them, we might find them. Just as we have already found meteorites from the moon on the Earth, and we found meteorites from Mars on the Earth, we could search the moon looking for meteorites from the Earth. And the really interesting ones would be the ones that are older than any current Earth rocks. Earth rocks dated 4 billion years ago, which might preserve a record of the Earth's crustal near-surface environment at the time that life was originating on the Earth, may be preserved on the moon. So here's a picture. This is from Apollo 11. This is Buzz Aldrin's boot. And here is a rock. Now, this rock will just be a moon rock, of course. But the point I'm making is that there will be places on the moon where some of the rocks on the surface, or perhaps buried in the soil, may be meteorites from the early Earth, and therefore they may preserve a record of the Earth's early crustal environment at the time life was originating on the Earth. They will be very difficult to find. There won't be very many of them, and I'm not saying that this is going to be very easy, but I am pretty confident that they are there. So if we explore the moon enough, then eventually we will find these potentially very valuable samples of the early Earth. And then the point about astrophysical processes. And now this is of interest to astronomers as well as astrobiologists, but some of these astrophysical processes certainly are relevant to astrobiology because they influence the habitability of the early Earth, or even the more recent Earth. So there are two astrophysical records which I think the moon may preserve for us, acting like as a time capsule, preserving evidence of the early solar system and galactic environment. So the first, obviously, you see the picture on the left is a large solar prominence. So large, which is probably going to give rise to a solar flare at some point. So the activity of the sun will affect the habitability of planets in the inner solar system. So we would like to know how common solar flares were in the ancient past. How active was the young sun? Because this would have affected the radiation environment of the terrestrial planets. So I'm going to argue the moon preserves a record of that. And then the picture on the right, this is a galaxy. So this is the spiral galaxy, Messier 51. But if it were our galaxy, then the solar system would be somewhere like there, where the red pointer is at the moment. But in the history of since the solar system formed, the solar system will have been around the galaxy, well, about 10 times relative to the spiral arms. I mean, everything is rotating, of course, but the sun is rotating around the galaxy faster than the spiral pattern is rotating. So relative to the spiral pattern, the solar system has been around the galaxy about 10 times since the solar system formed, which means from time to time it will pass through these things. These are spiral arms, where spiral galaxies like the Milky Way, the density of stars is much greater. The density of massive stars that explode as supernova explosions are much greater. And these are potential hazards for life. If the solar system was too close to an exploding star, then the radiation environment could be well, it depends how close we were, but could be very bad. And similarly, the dark dust lanes that we see in these images, I mean, these are dense interstellar clouds from which stars form. But as our solar system, as it's gone around the galaxy about 10 times, will occasionally pass through dark dust clouds as well. And these also could have effects on the habitability of the inner solar system because they will collapse the sun's heliosphere. and expose the inner planets to the interstellar radiation environment, whereas at the moment we're kind of protected by the sun's magnetic bubble. So I'm going to argue there's a potential record of the past history of the galactic environment of the solar system on the moon as well. And basically, they're going to be derived from records of solar wind particles implanted into the lunar soil. So solar wind particles implanted in the lunar soil potentially provides a record of past activity of the sun. And galactic cosmic rays, so these will be high energy charged particles, mainly protons, which, because the moon has no atmosphere and the moon has no magnetic field, these cosmic rays from the galaxy also strike the lunar surface and leave a record. So if the solar system had ever been close to a supernova, for example, we would expect to see a spike in the cosmic ray flux at the corresponding time. So I think the moon preserves a record for that, of that as well. But in order to explain why I think the moon has this record, I've got to do a slight digression into geology. So this is where astronomy and geology meet. If I go back to Buzz Aldrin's blueprint, we know that solar wind particles and the products of galactic cosmic rays are implanted into this soil, so it's called lunar regolith, because we've extracted them from Apollo soil samples that came back from the moon 50 years ago. But because of the way Apollo did its sampling, if you just scoop up some of this soil here, which of course the Apollo astronauts did, this from Apollo 11, this surface is about 3.8 billion years old. It's very old, which means that, yeah, we have extracted solar wind particles and galactic cosmic rays from this soil, but it's that what the record that we've got there is an average over billions of years. It doesn't have the time resolution. What we really want is something, a geological process that can tell us what the cosmic rays were doing 3 billion years ago, what the cosmic rays were doing 2.5 billion years ago, what the cosmic rays were doing 2 billion years ago. Some time-stamped cosmic ray and solar wind records, whereas all we have from our current samples of Apollo soils, and also the soils from the Soviet lunar missions and now from the Chinese Chang'e missions, they're all just collected from the surface. And therefore they just give an average cosmic ray flux, an average solar wind flux. But there will be places on the moon where the moon's own geological processes will give us a time-resolved record. So consider the picture on the left. Suppose there's a lava flow forms on the moon, of which there are lots. Micrometeorites coming in from space churn it up and produce a soil, a regolith. And then the regolith acts like a sponge, and it soaks up cosmic ray and solar wind particles. But then, if you imagine what happens at a later stage, I suppose another lava flow goes over the top. Then what's happened is that this regolith that was once on the surface is now buried. And so it doesn't get any cosmic rays or solar wind anymore. It's a trapped, buried regolith protected by an overlying lava flow. And meanwhile, the process starts again at the top. So I'm going to call this trapped regolith a paleo-regolith, an ancient regolith. And it will contain, this trapped, buried paleo-regolith will contain the solar wind particles, the cosmic ray particles, any meteorites derived from the early Earth, but all taken from a well-defined time horizon between the ages of the underlying lava flow and the overlying lava flow. So if you were to set up a drilling rig here and drill down through this, if you were to be able to date the lava flow 1, date lava flow 2, everything you find in the paleo-regolith in between would come from that particular time interval. And so it would be a time-resolved record of the activity of the sun and the galaxy. So here's a picture of, just to illustrate, this is a picture of part of the near side of the moon, Mare Imbrium. There's an old lava flow here, 3.3 gigayears old. There's a younger lava flow here, 3.0 gigayears old. These ages are estimated from counting craters on them. Neither of them have been sampled, but anyway, the approximate ages, that'd be good enough to illustrate the point. So 3 billion years ago, this lava flow here, I'm just tracing its outline, flowed over the top of this older lava flow 3.3 gigayears ago. 3.3 gigayears old, so 300 million years older. So if you wanted to know what the solar wind and the galactic cosmic rays were doing in the interval between 3.0 and 3.1 gigayears ago, you could set up a drilling rig there. This cliff face is about 50 meters high, so quite a big drilling rig. But anyway, just drill through the overlying lava flow. You would encounter a paleoregolith that's 3 billion years old underneath it, that's been developed on top of this older 3.3 gigayear-old lava flow. And so if you were to extract from that sample, that core sample, you would extract the cosmic ray and solar wind records of what the sun was doing 3 billion years ago and what the galaxy was doing 3 billion years ago. And then by repeating this in many other places on the moon with many different ages of paleoregoliths, eventually you could build up a history of what the sun and the galaxy have been doing over the last 4 billion years. Now, at this point, I would pause and ask whether anyone's got any questions. But I think since I'm giving this talk online, I'd better save the questions to the end. But it'll be obvious to you that, I mean, these records are going to be difficult to get to. It's going to require a lot of exploration on the moon. But if we ever in the position where we have a lot of people on the moon, operating perhaps on the moon in the same way we operate in Antarctica, for example, then these records are probably we will be able to access them, but it will be difficult, and we haven't accessed them yet. Oh yeah, so to access them, we're probably going to need to set up a drilling rig or something on the moon, and this becomes and one of the things that a future human exploration of the moon might be able to enable. Just go, so before I get to that, I just make one point, because conscious of the fact that the next talk is on Mars. Drilling on Mars, of course, would be even more interesting from an astrobiology point of view, because on Mars, we suspect in the deep subsurface there may be aquifers where water might still be liquid, and we'd like to know whether life exists in the Martian subsurface. But the point I want to make is that deep drilling on planetary surfaces is a technology we don't yet have. And I think the moon, we could develop this, if we develop this technology on the moon to access buried paleoregoliths on the moon. Once we've developed this drilling technology on the moon, of course, it will also be available for use on other planets, including Mars. Right, so I would have paused there because I'm about to switch gears now. And this is where you may think the talk is becoming a little less serious, but actually I'm quite serious about this. Strange though this may seem, but it's going to take a little bit of exploration. Sorry, it was going to take a lot of exploration, but it's going to take a bit of explanation. So one of the if like astrobiology, we want to know whether life is common in the universe, and we don't know. But if life is common in the universe, then some fraction of that life might have followed an evolutionary trajectory similar to our own and developed a technical civilization, technological capabilities. And some of that life, presumably, might be even more technically capable than our own present capabilities. So then the question is, we should search for it. And most of that is done with SETI, with radio telescopes. And although I don't have a slide on this, the moon can help even there, actually, because the far side of the moon is shielded from artificial radio noise generated by the Earth. So the far side of the moon is probably one of the best places in the solar system to do radio astronomy from, including SETI. So that's just an aside. But here I'm talking about something different. Suppose a technically advanced spacefaring civilization had come to our solar system millions of years ago or billions of years ago, I mean, the galaxy is very old, would it have left records behind? So this some of you may recognize, I don't know whether you've seen the movie 2001: A Space Odyssey. It's an old movie now, 1968, but if you haven't seen it, I strongly recommend seeing it. Where the movie is predicated on finding this extraterrestrial artifact, which is this obelisk, which had been buried on the moon. So this shows a trench that's been dug to expose this alien artifact buried on the moon. And I suppose we could look for such things. We should. But the main point I want to make about alien artifacts on the moon isn't macroscopic objects like this. It's a bit more subtle. And because it's a bit more subtle, it'll take me 2 or 3 minutes to explain. But don't worry, I'm looking at my watch. I owe this argument to a Ukrainian astronomer named Andrei Arkhipov, who was publishing it in the early 1990s. And his argument was, I think it's an excellent argument and it deserves to be better known. His argument was that if technologically capable spacefaring civilizations have existed in the history of the galaxy, then they would be generating space debris. We ourselves are already generating a lot of space debris. But his particular idea was if they get to the point where they start mining asteroids. So this is something that we are on the verge of being able to do. In the next few decades, probably we will start to mine asteroids. So here's an artist's drawing of a spaceship mining an asteroid. But Arkhipov's argument was if alien civilizations have got to the point where they're mining asteroids, they'll be producing a lot of debris. And some of this debris won't stay in their own planetary system. Some of the debris will be, especially the small stuff, the micron or submicron sized particles, will get blown out into interstellar space by the radiation pressure from their host star. And a lot of this mining debris will end up collecting in interstellar space. It will end up being collecting in interstellar clouds in between the stars in the galaxy. And the more technological civilizations that have evolved in the history of the galaxy, the more space debris there will be. Now, bear in mind, I said this relative to the spiral arms, the solar system has been around the galaxy about 10 times, multiple times anyway. So the solar system, if the interstellar medium contained small artificial fragments, artifacts, due to alien civilizations in the history that have existed sometime in the history of the galaxy, then some of this stuff will have flown into the solar system. So where would we look for it? The moon. I mean, there are other places in the solar system as well, but the moon is the closest such place. A planet with no atmosphere, a relatively quiet geology. This is probably where you might hope to find these alien artifacts if they exist. I mean, whether they exist or not depends on how common extraterrestrial technologies are, and we have no idea. I mean, there might not have been any. But Arkhipov's argument was that if there had been a lot of them, some of their space debris may flow into the solar system and we could look for it, and the moon would be probably one of the best places to search for it. So it doesn't really cost us anything to look. I mean, I know this argument seems a bit strange, but if we're going to the moon anyway, if we're going to go to the moon to look for ancient cosmic rays and Earth meteorites and water ice in permanently shadowed craters, I mean, we're going to explore the moon anyway. It doesn't cost us anything extra to keep an eye out for possible alien artifacts while we're doing it. So I'm happy to answer more questions about this if anyone's got any at the end. Then there's the more biological aspect of what we could do with astrobiology on the moon. Obviously, a lot of experiments have been done on the International Space Station. This shows one of these experiments where biological samples are exposed to the vacuum and radiation environment of low Earth orbit to see the effects of this environment on living things. But of course, low Earth orbit is not quite the same as deep space because it's protected by the Earth's magnetic field. So if we want to understand how biology might cope or survive in in deep space environments, then the moon is the nearest such place where we might do such experiments. And if we were to imagine setting up a moon base or something, I would anticipate there'd be a lot of experiments like this exposed facility this is on the space station, but you could imagine many similar experiments on the surface of the moon. But then, from an astrobiology point of view, there are a couple of other aspects I want to bring across, to try points I want to make. We would very much like to know how long living systems, say bacteria, basically, might survive in a lunar environment. And even if the bacteria don't survive very long, of course, they will break down into organic compounds. And how long would these biological compounds derived from broken down cells, how long would they survive? And the moon does provide a natural place, an obvious place to do experiments of that kind. And in fact, we've already unintentionally made a start. So this bag here underneath the lunar module, unfortunately, contains a lot of human waste from the astronauts. And it's been dumped there on the moon, which seems these days a rather environmentally unfriendly thing to do. But the point is, it was. So this material is there, could be sampled. In principle, we could go back to these places and we could see what has happened to what was certainly once a very rich biological sample after 50 years of exposure on the moon. A more subtle point is this shows an Apollo lunar module. This is Apollo 11 lunar module, and it's coming back from the moon. You can tell that because it doesn't have its landing legs. So when this picture was taken, Armstrong and Aldrin were inside here. So there were 6 of these successful Apollo landings on the moon. And astronauts stayed in these spacecraft for up to 3 days. And so by the time they'd finished, the insides of these spacecraft would also have been full of microorganisms. And then what happened? The astronauts came back to Earth, of course, but these vehicles, the ascent stages of the lunar modules, were crashed back into the moon. And they were crashed into the moon deliberately, not because it's kind of an act of vandalism, but because they provided data points for the seismometers that had been left on the moon. They're part of the geophysics experiment. By crashing these spacecraft back into the moon, they created a big bang, which then was used to trigger the seismic detectors to work out the structure of the moon's crust. But the point is, all of these millions of microbes left behind by the astronauts, they would have been in this spacecraft when it crashed. So if we were to visit these crash sites and see what survived, or maybe nothing survived, but see what organic molecules remain, this would provide insights into the extent to which life survives in an interplanetary environment, and therefore provide insights into planetary protection policies, and also the extent to which life might travel between planets, let's say within meteorites. So that's called lithopanspermia. By studying these samples left by the Apollo missions, we can at least see what effect the space environment has had on living systems exposed to the space environment for 50 or 60 years. So there's a lot of biology that one could do by going to these places, these landing sites, and seeing what is left biologically. Human physiology. So we certainly, with an eye to going to Mars, we could learn a lot of. This is another good movie. Maybe you've seen it, The Martian. There's a lot of science we could do on the moon if we have this kind of, and on Mars if we have this kind of capability. But before we try it on Mars, there's a lot we have to learn about how the human body would react to a living long term, for months or maybe a years on end in a low gravity environment. So Moon has 1/6 of a gravity. Mars has sort of almost 1/3 of an Earth gravity. We've done a lot of experiments on the space station. This is UK astronaut Tim Peake doing an exercise bike. We've got a lot of experience on what microgravity does to the human body, thanks to space station research. But what does prolonged exposure to 1/4 of a gravity or 1/6 of a gravity I mean, we'd like to know that before we send people to Mars, I think. And again, we could learn that. We could learn a lot of that by sending astronauts to the moon. And then once we're confident about how to prepare humans for living in low gravity and high radiation and dusty, I mean, there are a lot of hazards, dusty, high radiation, low gravity environments. We're going to need to learn how to do all of that when we send people to Mars, and sending people to the moon could help. So these are all my reasons for wanting to explore the moon, and well, not all of them. So 2 more slides. Just I've got 5 minutes left. So yes, 2 more slides. Firstly, I'm not trying to convince you that astrobiology is the only reason for wanting to explore the moon. I hope I've made the case that astrobiology would learn a lot by exploring the moon, but it's not the only reason for exploring the moon. Even for other scientific reasons, there's a lot of planetary science to do on the moon that's got nothing to do with astrobiology. I mean, the moon is an interesting planetary body in its own right, so there's a lot of lunar science that we'd obviously do. Here is an artist drawing of some astronauts back on the moon doing some geology. In the more distant future, the moon may contain resources which may be of economic benefit. Certainly there'd be of economic benefit for activities on the moon, like if we wanted to build a moon base, it would make sense to use lunar resources. It's conceivable, like I mentioned, mining asteroids, it's conceivable that we may find economically useful materials on the moon. I mean, it'll be a long time before anything gets to this scale, I think. But still, we should maybe keep an open mind to it. But bear in mind, there will then be tensions. If we're going to mine the moon for its resources, there will be then a tension with the scientific aims of wanting to preserve the lunar environment for scientific purposes. So we wouldn't want a big mine like this in the same permanently shadowed crater that we were looking for ices derived from comets in the early solar system, because we wouldn't want those ices to be contaminated. So there is a future space policy issue here of how we can both preserve sites of special scientific importance on the moon and, if it happens in the future, find places on the moon where space resources could be utilized. I mean, this is a discussion that is starting now at the United Nations, but it needs quite a lot more thought, how to preserve science, how to get both science and commercially valuable activities working on the moon in parallel so they don't interfere with each other. And then I've already made the point that the far side of the moon, at least, is an excellent place for radio astronomy, including SETI, but not only SETI. There's a lot of mainstream radio astronomy could be done on the moon as well. I would just, as an aside, just say that these are the scientific reasons for wanting to return to the moon. I do think there are a lot of societal and cultural reasons for wanting to send people to the moon and to Mars as well in the future. So I know this isn't the topic of your conference, so I'm not going to say too much. But what I would say is this: I think the future of space exploration provides a potential activity that the whole human race might be able to get behind. I think at the moment, if we look at the state of the world, we could really use a few things that are going on that unite us, in addition to all of the things that are tearing us apart. And I'm not saying that space exploration is the only such thing. It's not a panacea, but it is a thing. I think an rigorous international space program could help unite humanity in a way that would be. I mean, it's not suddenly going to make everything perfect, but it would help. Anything that pulls humans together instead of driving us apart would be beneficial, and I think a human space program could help with that. But that also requires a lot of thought and a lot of international consultations. So anyway, this is my last slide. I hope I've convinced you that the moon has quite a lot to tell us about. It's astrobiologically interesting. But it's quite large scale activity, right? If we're talking about drilling down for paleoregoliths, exploring craters at the poles of the moon for ices, searching for Earth meteorites, doing biological experiments, setting up radio telescopes. There are multiple sciences that could be done on the moon, but they would require quite a lot of scientific infrastructure on the lunar surface to support all of this activity. And I think the analogy, I made a reference to it earlier, I think the analogy would be like the way we explore the continent of Antarctica, where on Antarctica we have lots of sciences, lots of science being done, atmospheric science, geology, biology, zoology, meteorites being collected. All of these sciences being done on the continent of Antarctica, but they're all supported by a network of permanently inhabited or semi-permanently inhabited scientific outposts, like the McMurdo Station or the Amundsen-Scott Station, the Concordia Station, the Vostok Station. So we have these research outposts in Antarctica, and they facilitate a lot of science on Antarctica. So I think we need something similar on the moon. I think eventually we may need something similar on Mars. But so here's an ESA drawing of a rather ambitious moon base. It'll be a long time before we have anything on quite this scale on the moon. But I do think something like this would facilitate a lot of scientific research on the moon, much of which will also be relevant to astrobiology. So thank you. I'll finish there, and I think I've got a few minutes for questions.
Thank you so much. And if my moderator friend could read the Turkish version.
Sayın Profesör Ian Crawford'a konuşması için teşekkür ediyoruz. Kendileri adına tema vakfına fidan bağışında bulunduk. Bağış sertifikasını e-mail üzerinden ileteceğiz. Şimdi 10 dakikalık soru bölümüne geçiyoruz. Katılımcılarımızı sorularını sormaya davet ederiz.
We extend our sincere gratitude to Professor Ian Crawford for his presentation. We made a donation of saplings to the Turkish Foundation of Combating Erosion, Deforestation, and the Protection of Natural Habitats on their behalf. We will send a certificate via email. And we now have 10 minutes for questions, and we encourage our participants to ask any they may have.
There is a question from the chat. I will read it to you. What would be the best strategies to distinguish extraterrestrial microorganisms or alien artifacts from terrestrial origins in Moon's records?
Yeah, that's an excellent, a very important question. So I think if we were to take alien artifacts, so Archipov's idea was that we might find fragments. So if aliens have been doing a lot of activity in their own solar system, even if they haven't deliberately come to us, but they produce debris in their own solar system, which has then been spread out into space, and the moon might collect it. He was thinking that we might find fragments of exotic metal alloys, so whatever they built their spacecraft out of, exotic aluminium, titanium alloys, or other exotic materials. But then, of course, yes, we are polluting the Earth with we are polluting the moon with lots of artificial alloys and materials that we have made. So how would we tell the difference? So that's an excellent question. I think we would tell the difference through the isotope ratios. So whatever metals or things that we make our Earth artifacts out of, they will have a terrestrial isotope composition. Whereas any aliens that have originated around different stars in different planetary systems, almost certainly the main elements are going to have slightly different isotope ratios because they've come from different planetary systems. So I think we would search the isotope ratios to determine that they were alien origin or not. Now, for life forms, it's different. I don't think we're expecting to find any alien life forms preserved on the moon. But in general, I do think biologists have a technique. I think life that originates not on Earth is probably going to be biologically different from Earth life, even if it's basically carbon-based, even if it uses amino acids and proteins and lipids. which it might not. I mean, it might be totally different. But even if it's carbon-based, like our kind of life, its biochemistry is probably going to be different. I mean, its genetic code might be different. It might have 17 or 25 amino acids instead of 20 in its biology. I would suspect that we see biological differences that would enable us to determine, molecular biological differences that would enable us to determine that it's not Earth life. I mean, bear in mind, all Earth life, as we understand, has a common origin. And that's why we all have a common genetic code and a common underlying biochemistry. And an alien life would not, because it would have had an independent origin. So I think the biologists could probably tell the difference.
Thank you very much.
And I have a question for you. Given that some meteorites, like the Murchison meteorite, contain amphiphilic components and amino acids, hypothetically speaking, could space debris produced by space mining carry these elements of life to planets with water and potentially initiating some form of life? And additionally, do lunar meteorites also contain these components?
So there are two questions there. So I think in principle, anything that spreads organic material around, then potentially, yeah, if you introduce organic material, even it's abiotic organic material, to a planet that doesn't have any, then yeah, potentially you could kickstart a biology. I mean, I suspect it's not as simple as that, because all you're doing is providing the raw materials, and you probably need lots of other things as well as just the raw materials. But still, you could be providing raw materials for life to a place that didn't have it. So that could be a start. The second question, that's an excellent question, but so far, no. I mean, all the lunar meteorites we have are igneous rocks, and they just represent, they're just samples of the moon's crustal rocks, which don't contain organic compounds, because the meteorites that do, as you correctly noted, are the carbonaceous chondrites, like Murchison and Iendi, and these are the carbonaceous chondrites. So those have come from the outer solar system, and yeah, they are very rich in organic molecules. And they may well have played a part in introducing organic molecules to the early Earth at a time when life was getting started on the Earth. But the actual meteorites we have from the moon, no, they're just lunar igneous crustal rocks, and they don't contain organics.
Thank you. Do we have any more questions?
We have one more. Considering the potential for space mining to increase space debris, what are the most effective strategies or technologies we should develop to ensure sustainable space exploration while minimizing the environmental impact on the space ecosystem?
Yeah, so that's another interesting and important question. I think there are two aspects. I mean, we can think of technologies that mitigate space debris production as much as we can. But I think perhaps the bigger part is not so much the technology, but the politics. What we really need are strong international agreements on space debris mitigation. And currently we don't have any. I mean, like I mentioned, there are a number of discussions ongoing at the United Nations at the moment to try and reach international agreement on mitigating space debris, and including debris and on the moon and elsewhere in the solar system. And I think these discussions are at a pretty early stage. But it's it's like global warming, right? I mean, if you just stop carbon dioxide pollution of the Earth's atmosphere, it needs to be in a global agreement, because otherwise there's no point, because some countries will agree and others just won't, and therefore the atmosphere gets polluted anyway. And it's similar with space debris. We would need to negotiate a globally binding international framework for mitigating space debris and damaging environmental impacts on other planetary surfaces. Once we've agreed to do it, I mean, I think the technology is probably not the most important, it's not the most difficult bit. Once we agree not to damage extraterrestrial environments, I'm sure we can develop the technical means to prevent damaging environmental impacts. But we have to have some agreement at the political level to make sure that everybody, to define the rules and to make sure that everyone agrees with them. And that's an ongoing process. It's barely started. I mean, it needs to start, it needs to get going because it is important. So thank you for raising it.
I think that's all from the questions.
Okay.
If our technical team can cut the broadcasts.
Ben devam edeyim istersen. Değerli konuklarımız, bir sonraki oturum için 10 dakika ara verilmiştir. Son oturumumuz saat 15.10'da başlayacaktır.
Esteemed guests, a 10 minutes break precedes the fifth session. Today's last session will begin at 3 p.m. Additionally, you can participate in our giveaway that will take place at the end of the day by filling out the form shared in the chat. To confirm your attendance, you can access the participation form via links shared in the chat and during breaks.
Ben de aradaki kısmı söyleyeyim. Ayrıca chat kısmında paylaştığımız formu doldurarak gün sonunda gerçekleşecek çekilişimize katılabilirsiniz. Oturuma katıldığınızı belirtmek için de chat kısmında ve aralarda linklerden katılım formuna ulaşabilirsiniz. Okay. değerli misafirlerimiz, şimdi Sayın Profesör Franco Ferrari ve Profesör Doktor Eva Shushkuvi'yi Mars'a gitmeden önce tardigratlar uzaydaki diğer organizmaların korunmasına yardımcı olabilir mi? Konunun sunumunu yapmak üzere davet ediyoruz.
May I now invite Professor Franco Ferrari and Professor Dr. Eva Shushkevich to present their talk on Before Going to Mars, Can Tardigrades Help in Protecting Other Organisms in Space?
Thanks a lot. Welcome, everybody. It's very nice to be here. Thanks for invitation. And we would like to present today our experiment, the yeast tardigrade gene, with a full name as the title of our talk, Before Going to Mars. And I would like to say that Ian, in the previous talk, make a very nice introduction to what we would like to say, because I mean, we will just concentrate on one particular aspect of whole interesting things he was talking about. So this is a one talk divided in two parts. So this is one really talk together. And we are founders of the Center of Astrobiology and Related Topics in Poland. And we work at the University of Szczecin at the Institute of Physics. That's what you see on the picture now is the original patch of our experiment. So the heart of our experiment is a gene of a tardigrade represented by the double helix of DNA shown on the left. And the home of tardigrades is the Earth. So you can see this blue-gray background on the bottom of the picture, and that's our land, our planet. And the home of tardigrades is the Earth. And this you can see one of these indestructible animals shown in gray and black on the Earth. The yellow stuff in the flask, you can see on the picture, it is the yeast and the yeast cells, and they will be one of the hero of our experiments. And they are placed on the low Earth orbit where the experiment will take place. So you can recognize also in the picture the Moon and Mars, but about this I will talk a little bit later. So our experiment is the part of the bigger mission, which is called INIS. It's from Latin fire. And this is the first Polish mission to the ISS. And what you can see in the middle of the picture is one of the members of the crew, and this is Sławosz Uznański-Wiśniewski, the project astronaut of ESA, a Polish astronaut. So he will take care about our experiment and also 12 other experiments prepared by Polish space industry and research and teaching institution in Poland. So we are very excited about this, and there's a lot of things going on. Some of these things we will present here to you. And let me first say who we are. This is, of course, the whole interdisciplinary team from 3 different academic institutions in Poland. I am a coordinator of this project, and we work together with Franco, taking care of the whole aspects of the project. I mean, I'm an astronomer, astrophysicist, and astrobiologist, and Franco is astrobiologist and physicist, and we have in our team biologist, which is inevitable, I mean, to join the forces in this very complex endeavor, and which is sort of focused on the survivability of the organism in space. So that's very important. So we have here people from Adam Mickiewicz University in Poznań, Hanna Kmita and Andonis Karahitos, Nina Antos-Krzemińska, Łukasz Kaczmarek, and from University of Silesia, Izabela Poprawa. And the whole project is done in collaboration with Polish Space Agency, Polsa, and also European Space Agency, ESA. So let me put our experiments in the wider perspective. In fact, I will present the scene. I prepared a scene for Franco to then let him tell you more about the detail of the experiment. And our aim is to explore our home, to know what we can find there. And our home is our solar system. So we have 4 terrestrial type planets: Mercury, Venus, our planet Earth, and Mars. We have 2 giant planets, Jupiter and Saturn, and 2 icy giants, Uranus and Neptune. There are other important bodies in our solar system, like asteroids that or dwarf planets, as we call them now, Ceres, and Pluto with its moon Charon. And this is what we would like to explore. And there is a question to be asked. I mean, are there any sign of life in this particular planetary system? We hope to explore other bodies and to find the answer. So let me show you a few slides about how the solar system explorers are presented nowadays. I mean, what is the all different missions which were sent, which are planned to be sent to different bodies of our solar system. And I hope that the program of ESA will be something which will put together all us, the humans living on Earth, in the exploration of our neighborhood. And of course, I mean, there's a lot of action going on. I am not going to talk about all of them. I will focus on the very limited region here, Earth, Moon, and Mars. That will be the topic of our discussion today. Of course, from an astrobiological point of view, there are a lot of interesting sites outside these 3 bodies. There's a lot of attention nowadays to Venus. There are a lot of attention to the giant planet satellites. And you will hear about this in the near future. But let's come back now to Earth, Moon, and Mars. So that's the master plan of ESA, the one which involves these 3 different level of exploration. So the first one is, of course, low Earth orbit and International Space Station. And in fact, that's where our experiments will take place. But this is just the first step. What we are aiming for, we are aiming to expand it to the different environments, which Ian described very nicely in the previous talk. And we will be interested in the experiments on the new endeavor, which is Getaway to the space station on the orbit around Moon. And of course, Moon itself will be interesting as well. And then the next step is Mars, and we are trying to explore the possibility about this, the most exciting sites which we are focusing now on. And there is a Rosalind Franklin mission, which is the astrobiological mission, is going to explore different aspects of Mars environment. And do not forget about the sample return mission, which is also planned to take the pieces of Mars into our terrestrial labs. And that's the summary of what we are aiming here in our, step-by-step exploration to know what is around us, and are there any condition for life to exist or to develop, or maybe there was a life there before the time we are exploring. So here is the Earth, here low Earth orbit, and next is the Moon, and of course the Mars. And how are we going to go for information which we are interested in? So this is one of the programs of ESA, which is telling us what we are planning for the near future. So there's a humans in LEO, low Earth orbit, and humans beyond that. But for the moment, in this particular program, Mars and Moon will be studied by robots. So this is the humans and robotic missions, which are now developed and are in progress. So there is whole science concerned with this, and science in the space environment, and we are part of this particular program. And of course, what is here important to stress, the technology which must go on hand by hand with the exploration and with the scientific understanding. So our experiment is going into this category, the humans in LEO. Franco will tell more about it, but there is a connection with what we are talking about and what we are planning in our experiment. We are not making experiment with humans, but Franco will tell more about that. So that's how the scenery looks like. You can see here the distances and time delays between the different location and how much time you need to get information from there. So I put here also the patch of our experiment to let you know what is the meaning of the Mars and Moon, which is placed on our patch. Those are the next level of our exploration, going to the different, more astrobiological interested sites. So the Mars and Moon. And now I will tell just briefly what is the scope of our experiment. That's the title is microgravity, and because on ISS, as Ian already mentioned, we are still protected from the cosmic rays flux, which is by magnetic field. So, which is another important ingredient of, space conditions, which we should understand, and will be, and we need to sort of study the effects of these particular conditions. So the scope is to examine if a tardigrade gene, which is expressed which is expressing the mitochondrial protein, which is called alternative oxidase, can protect budding yeast from negative effects of microgravity. And you will hear about all this from Franco, but I will just say what are the fields of relevance of this particular experiment. So, I mean, there is a connection between what we are doing with the fundamental research and also applied science. So astrobiology, biochemistry, biophysics, biology, biotechnology, and medicines will, benefit from the results of our project and of the methods which we are using, in the plans in the future. And as far as applied research is concerned, so the space exploration, development of the space industry, food production, and energy. So this was the first level, and on the low Earth orbit, and Franco will describe the details. But I mean, we don't want to treat it as just the end of our studies, because we are interested in more extensive environments. So, I mean, we are interested in microgravity, but also ionizing radiation, cosmic rays. And for doing that, we need to develop technologies which will allow us to make experiment in the long term. So we need to feed our organism during the experiments. And for this, we need to make the automatic sort of laboratory, which will do what we want without the humans' interaction. So lab on chip, that's our next step. There's a lot of things, technologies which were developed, and you can see here the example of BioSentinel 2020, and there is a progress. Technology is getting better and better, and there is the chip, which is so small. You can see these fingers here, and then so you know what the miniaturization of these systems miniaturization is. So this is the future is going into the lab on chip and going to the microfluidic systems, which is necessary for biological experiment in space. And then the next step is Mars. So the most, this is our target. There's a lot of things to do in between. And in fact, Moon will be the one step in between. And we think that, proceeding this way, we can learn more. We can be able to prepare the flights which last longer than just flight to the moon. And we can have the possibility to provide food and energy for such trips, and also for the habitat, maybe in the future, on Mars. So I will thank you for your attention, and because we organized this talk just as one integrated talk, so I I will expect that we will take the questions at the end of Franco's talk. Thank you very much.
Okay, so let me take over. First of all, thank you very much. I feel honored to talk in this conference, at this conference, and Eva made already a great introduction to what I will talk now. And so I hope that my slides are visible for you. And oh, there is some little disturbance anyway. So while it is now under debate if it is if it is reasonable to send human missions into space instead of making robotic mission, or to spend this money to help the world on the Earth where it is necessary. I would like to. I have no time to discuss that, but some food for thinking. I would like to address you the experiment of John Calhoun in the 70s, where the population of the Earth were about 3 milliards. Now we have over 8 milliards, which made an equivalent of human societies in which the progress, the technological progress is very advanced, so that all problems of the society are solved, apart from the lack of space, and not only the lack of space, I would say, but an environment in which the contact between mice are increased, are multiplied, and that the results were catastrophic. So, so this is one thing that I want to point out for you. And the second thing is that now space exploration has started now for many years. And however, since 10 years, the private sector has joined in. And well, the first who started to be interested in the private exploitation of space was Robert Zubrin, space visionary. Just he was the right man, but at the wrong moment, because at that time in the 80s, when he was mostly active, the space exploration was managed mainly by space agency, national space agencies like NASA or ESA. However, again, food for thinking, I want to point out that there are very different visions about how to go to space. And in particular, I want to focus on two different visions, that of Elon Musk, the owner of SpaceX, in which the idea is colonize the massive bodies of the solar system, like, for instance, the moon or Mars, against the vision of Jeff Bezos, which is the patron of Amazon. And the idea here is to send a space station in the low Earth orbit. So in the neighborhood of the Earth, where we are safe from radiation, relatively safe. But of course, there is the problem of microgravity. And indeed, these are the main stumbling block on the path to space for humanity. One is the psychological stress, which is not to neglect, because well, the body's function in the absence of gravity, first of all, are very distressing, and there is a remoteness sensation, loneliness sensation, and for instance, it is difficult to sleep because, for instance, the astronaut from the Apollo mission were seeing flashes, probably due to ionizing radiation in the eyes, and this was disturbing. Of course, there is also ionizing radiation. If you want to go to Mars, deterministic effects, which are those effects that occur when the radiation is so high that the tissues are destroyed, and then organs stops working. So these deterministic effects are negligible, because the doses, for instance, in a typical missions to Mars are about 1 sievert or even less. But however, there are stochastic effects. So this means long-term effects that are completely random, like the like the appearance of solid tumors or blood tumors. So these are not negligible. And we are adapted to some extent to low-level radiations coming, for instance, from photons and muons. And we had, together with Professor Shushkevich, an hypothesis that this low-level radiation was actually shaping the evolution of the organism on the Earth. However, in the space, there is high-level radiation, and in particular, very heavy and very energetic particles like ions, heavy ions, which can change the game when going into space. And of course, there is a microgravity, which is also the source of several health problems that the astronauts have. In particular, muscles are getting weaker. There are also problems in the circulation of blood, because a little bit our organism is confused by the lack of one privileged direction. And in particular, we will see that also microgravity has effects which are very similar to ionizing radiation. So they generate something that are called the reactive oxygen species. So what is our tardigrade gene experiment? Eva has already talked about that, so I will skip the fact that we are a consortium of 3 universities. And the idea is to enhance the adaptability of yeast. So this is budding yeast, a commercial yeast that can be bought commercially online, for instance, to microgravity condition in space by editing the genome of the yeast using the CRISPR-Cas9 method. Which is now, I would say that this method has, of course, can introduce mistakes when changing the genome, but the mistakes are very small, and we use a particular substance that kills the cells that have this Cas9 protein, and what is left, what it is leaving, are just the cells that have been correctly changed. So that there is no problem of mistakes. And then the idea is to evaluate the influence of microgravity on the functionality of mitochondria. I will talk about that in a moment, and on the vitality of microorganisms. And then, of course, we have in mind biotechnological applications of yeast in space. This will be in the conclusions. So why mitochondria? Maybe some of you know the Krebs or citric acid cycle. And this is the way in which cells produce energy to maintain their functions. And essentially, this is a cycle of chemical reactions and not only, in which the ATP, so adenosine triphosphate, is giving energy by losing one of the phosphate group that you see here. So this is the adenosine triphosphate. That there is one molecule of adenine. Then you don't see that, but this is a sugar called the ribose. And here there are the 3 phosphate groups in the tail. When one of these bonds with the last phosphate is lost, then this molecule generates energy that can be used for the functions of the cell. And instead of 3 phosphate groups, we have just 2. And this molecule is called ADP, diphosphate. Here there are also applications to, for instance, ketogenic diet, which I don't suggest to do if, so this is for getting thinner, but I don't suggest it without the support of a dietologist or a physician, because it can have some effects so the heavy effects for the body if it takes a lot of time. So what's the matter is that the Krebs cycle consumes a molecule called acetate in the form of acetyl coenzyme A, and it reduced this nicotine amide adenine dinucleotide, NAD, in a form that here is charged, because you don't see here, but this is not H. I don't know why I have this. I don't know. Anyway, sorry. So there is so essentially, this is charged because it has lost an electron, and is at the oxidized form. And the NAD H with an hydrogen atom at the end is the reduced form of NAD. And this is used to gain energy, because this NAD H wants very badly to donate electrons to other molecules. And when it does that, energy for the metabolism is gained. And sugar, fats, and proteins are used to produce this molecule that starts the process, acetyl coenzyme A. And in the ketogenic diet, the input of carbohydrates is reduced in order to reproduce the situation of a starving person, in which the main source of this acetate are fats. So one is the weight is decreasing because that person is consuming their own fats. However, this process, as I said, is dangerous. So now let's forget the Krebs cycle, and just have in mind this NADH molecule, which is the reduced form of NAD. And let's see just what happens in the mitochondrion. This is the organelle that is the powerhouse of the cell. And this organelle has 2 walls, an outer membrane and an inner membrane. In the middle, in the interstitial space between the 2 membranes, there is an excess of proton. and in the inside of the inner membrane, there is a low proton concentration. Here you can see with the letter M the size of the mitochondria. They have also some astrobiological importance because it is thought that eukaryotic cells inglobated some smaller organisms, and these organelles are coming from such a smaller organisms, simpler organisms. However, the organelle by itself, is not able to live outside of the hosting cell. So let's see what happened here, how this energy is gained. So first, as I said, so this is the outer layers made by lipids, and this is the inner layers. In the middle here, there is an excess of protons. So protons are hydrogen atoms without an electron. So this flow of protons from the intermembrane space, where hydrogen atoms, ions are more, to the inner part, makes here this coenzyme, this protein, transform ADP into ATP, so that this is like recharging batteries. However, when doing this, of course, there is a flow of positive charged particles in the inside. So one should find a mechanism to again restore this difference of charges. So here there are proton pumps. So here we have 4 proteins that they are acting like pumps that take electrons and put them outside. They take electrons and they take the electrons from this NAD H and starts an electron transport chain in which this electron of the NAD H molecule is transferred from one to the other of these molecules. And this generates a chain of reaction, reducing reactions that gives energy to these proteins. And this energy, by a mechanism that, as far as I know, is still unknown, pumps these hydrogen ions, so the protons, side of the inner part, so to restore the gradient of charges needed by this big protein to transform ADP in ATP. And of course, here we see a lot of charges is around, and these so molecules that are charged make problems because, for instance, radiation acts in the same way. And there are, of course, radiation that destroys the cells, the DNA, but are also radiation that ionizes molecules inside the cell. And these ionized molecules are charged molecules that interact very easily with other molecules, and they are making problems. So whenever there are stress conditions, there is the production of reactive oxygen species, and this number of reactive oxygen species can grow considerably. So in microgravity, this is actually the case. So what is our answer to this problem? So we use tardigrades. We focus on tardigrades because they are extreme tolerant organisms. They are extremely resilient. They are less than 1 millimeter, but can live for several years. And they have a mechanism called cryptobiosis, in which they completely dehydrated and stop their metabolism when the situation is very bad. For instance, there is no water because they need water to live. And in this cryptobiosis state, which is specifically in the case of tardigrades called anhydrobiosis, because they are without water, they are completely desiccated. They can survive in extreme conditions, as I showed here, minus 272 degrees Celsius, and for some time they can live even if the temperature is 130 centigrades. And in the lack of oxygen, vacuum, ionizing radiation, microgravity, and high pressure, the vacuum has been already studied in the case of the ISS experiments, and it has been shown that for days these tardigrades could survive in the vacuum outside of the International Space Station, and then without problems they start again to live and reproduce themselves. Here we focus on a particular protein of the tardigrades, which is the mitochondrial alternative oxidase, AOX, which enables mitochondrial reduction oxidation reactions. And they are important in order to adapt the tardigrade to variable biotic and abiotic stress factors. So what we will do, so we take the strains, so the strains of yeast, and we replace one gene that it is used to get energy from galactose in yeast, and we replace it with the protein AOX of a tardigrade species that it is called Milnesium insectum. Now, in the presence of glucose, so here we see that we have cultures, 120 cultures that will be sent into space. And there are two kinds of medium. So the culture medium is of two kinds. The YPD medium contains glucose. In this case, the GAL1 gene is suppressed because the cells uses glucose in order to get energy. And this gene is not used. However, in the case of the YPS medium, there is no glucose. And in this case, this galactose 1 gene, GAL1 gene is activated, and however, it is missing. So instead of the GAL1 gene, the tardigrade gene is activated. And this gives, so what is the effect of this gene? So we see here again what I showed before. So this is these are the molecules that are used. So 1, 2, 3, and 4 are the proteins that are used to pump out of the inner membrane these protons. And this route that I showed before of the electron transport chain is now modified by a small bypass in which a part of the electron is deviated to this AOX protein. This makes this tardigrade protein, this makes the yeast cells more resistant, and in particular, it regulates the number of charged molecules, this reactive oxygen species. And we have already experiment made on the Earth without, of course, microgravity. that shows that the protective role of the tardigrade AOX is working. In particular, here there is one strain of yeast that is lacking the SOD1 gene. So the delta means delete. And in this case, the strains becomes very weak against the production of this reactive oxygen species. But with the addition of the AOX tardigrade protein or gene, they survive and they are able to multiply as the native strain. So let me go to the conclusion if I have still time. So the aim of our experiment is to solve at least partially the problems related to the harsh space condition affecting organisms at the basis of the food chain like yeasts. And now the yeast strain Cerevisiae that will be investigated is very well known because this is budding yeast, so a kind of yeast that is used to make cakes. And for instance, here there is a kind of Polish yeast plum cake, which is called the drożdżówka. And moreover, the yeast shares with humankind 30% of their genome. So it is not a wonder that yeast is very popular among researchers working in space science. And finally, yeasts have a double kind of respiration in which energy can be, which is different if there is oxygen or if there is there is an excess of CO2. And if there is an excess of CO2, then the cells starts to, so in the absence of oxygen, the cells start to produce alcohol, which is called ethanol, that can be used as a biofuel. And here in the picture, there is this biofuel generated by yeasts. And of course, there is also some aspects related to the health of humans, because as I said, the yeast is a practical model to study the effects of the cosmic environments, like for instance, microgravity on the health of astronauts. And about the health of astronauts, we have other plans based on organoids. And we have a preprint about that. And however, this is another story. So thank you very much. Well, I am Professor Szuszkiewicz. We will gladly answer your questions.
Sayın Profesör Doktor Franco Ferrari ve Profesör Doktor Eva Szuszkiewicz konuşmalar için teşekkür ediyoruz. Kendileri adına Tema Vakfı'na fidan bağışında bulunduk. Bağış sertifikasını e-mail üzerinden ileteceğiz. Şimdi 10 dakikalık soru bölümüne geçiyoruz. Katılımcılarımızı sorularını sormaya davet ederiz.
We extend our sincere gratitude to Professor Franco Ferrari and Professor Dr. Eva Szuszkiewicz for their presentation. We made a donation of saplings to the Turkish Foundation for Combating Erosion, Reforestation, and the Protection of Natural Habitats on their behalf, and we will send the certificate via email. We now have 10 minutes for any questions, and we encourage our participants to ask if they have any.
We have a question. I will read it for you now. What future experiments experiments are planned to further investigate tardigrade resilience in space?
Yes, so one of the the simplest outcome will be just to count the growth of the cell, their weight, and this will say which of these strains has better adapted to the condition, to the space condition. Another experiment is using a fluorescent protein, which is coming from a jellyfish, A. victoria. And in this case, this is to rule out that there is some better adaptation just due to the fact that we have cancelled, erased this, deleted this GAL1 gene. And instead of this gene of the tardigrades, there will be this fluorescent gene of the jellyfish, and it will be seen how much this medium without the glucose has produced. So how much this gene that it is missing but has been replaced has been expressed. And this will show that, and we have already a proof on the years that there is no bias due to the fact that this GAL1 gene has been deleted. And then there are other experiments that are a little bit more looking at the genome and of the yeast cells. So we have this in plan. So the most difficult part is to distinguish, because the experiment will be performed in the space and also on the Earth. So the problem is that in the ISS station, the radiation is quite high because it is actually of the same kind of the level of radiations on the surface of Mars. So how to distinguish the effects of radiation from those of microgravity? So this is the basic problem. And the idea is to irradiate the yeasts for a long time, more or less giving the same radiation on the Earth to see what's the difference, what is generated by microgravity and what is generated by radiation. And for this, we think because the access to these facilities in which generate radiation is very tough, and we need something that it is of the order of 28 days. So 1 month in which the irradiation should be steady. So we thought to have some material that is like cobalt, for instance, that it is radioactive, and without using so many facilities that are used for other purposes, to irradiate for a long time enough our samples.
Thank you so much. I also have a question. Are there genetic or molecular insights from tardigrades that could be applied to other organisms for space survival? I wonder.
I don't know if Eva wants to answer to that.
No, go ahead.
So of course, so here, so this is a very good and also difficult questions, because of course, yes, because so there are many organisms which are very simple, and there is no problem to add at some point with this CRISPR-Cas9 technique, this tardigrade gene, and since this cell respiration mechanism is basic essentially in all organisms. Bacteria have something different because they don't have organelle, they don't have mitochondria, but it is something very similar. So there are a lot of organisms that have the same mechanism of respiration. So this technique can be applied, of course. It will not be applied to humans because, as I said, CRISPR-Cas9 technique can give very nice results, but also can introduce genetic manipulation. So if we do something in vitro, then there is no problem because the cells that are not fine, they can be destroyed. But in people, of course, this is a problem. Anyway, let me say that this CRISPR-Cas9 method has been already used. So there is a disease that affects the Mediterranean country, and there is sickle blood cells or red blood cells. I don't know if in Turkey there is something like that. In Italy, for sure. And in that case, the red cells are in the form of a sickle. They should be spherical, but in that case, they are not. These people have some advantage, so some protection against certain diseases. So this evolution mechanism is working, this natural selection. However, they have a very big disadvantage that usually they don't survive adulthood. So when they are about 20 years old, they unfortunately die because this is a serious disease. In that case, these methods are, I think, allowed because, and indeed, they save life. So we are not only able to make wars, for instance, so bad things, but also to make miracles, because until now this disease was killing. Luckily, it is rare, but the people affected were dying very, very soon. But now they can live and become old and so without problems. So this is a miracle.
Thank you so much for your answer. Is there any question in the chat?
Let me see. No, all the questions have been asked.
Too difficult. It was difficult to explain everything like the Krebs cycle, and also something is missing because this CRISPR-Cas9 method, this explanation is missing. So, but there was no time for that.
All right.
Okay, then. That's all, I guess.
Okay.
Can you read the Turkish
Okay.
sentence?
Ekran başından ayrılmayın. Kısacık bir aradan sonra çekilişimize devam edeceğiz.
Don't leave your screens. We will continue with our giveaway right after a short break.
Değerli katılımcılar, şimdi çarkıfelek çekilişimizi gerçekleştireceğiz. Bu çekilişin kazananları 2 kişiden oluşacak ve ayrıca 3 yedek isim belirlenecektir. Çekilişimiz yalnızca Türkiye'den katılımcılar için geçerlidir. Eğer isminiz okunursa Team Origins of Life Instagram hesabı üzerinden bizimle iletişime geçebilirsiniz.
Dear participants, we will now proceed with the Wheel of Fortune raffle. The winners of this raffle will consist of 2 individuals and 3 backup names will also be drawn. Please note that this raffle is only for participants from Turkey. And if your name is called, you can contact us via Team Origins of Life Instagram account. Tamam. Tamam. Esma.
Kim diyecek misin?
Berfin. Congratulations to the 2 winners. Let's just draw the 3 backups. Oh, Özge. Özgür Timuçin Kutlu. And the last person. I don't know why, but there's 2 Esmas there. Gülistan Kavak.
Kazananları tebrik ederiz. Instagram hesabımızdan bizimle iletişime geçmeyi unutmayın. Konferansa katılımınızı kontrol ettikten sonra hediye kodunu sıralaştırmak için gerekli işlemleri yapacağız. Yıldız Teknik Üniversitesi Biyoteknoloji ve Genetik Kulübü olarak bu sene 4.sünü düzenlediğimiz Astrobiyoloji ve Yaşamın Kökenleri konferans etkinliğimizin ilk günü sona ermiştir. Bizleri onurlandırarak davetimizi kabul eden bütün konuşmacılarımıza ve siz değerli katılımcılarımıza teşekkür ederiz. Son oturumun katılım formunu birazdan paylaşacağımız linkten erişebilirsiniz. Gelecek etkinliklerimizden haberdar olmak için sosyal medya hesaplarımızı takip edebilirsiniz. Yarın etkinliğimizin 2. gününde görüşmek dileğiyle. Sağlıkla kalın, bilimle kalın.
Congratulations to the winners. Don't forget to contact us via our Instagram account. After verifying your conference participation, we will proceed with the necessary steps to send you your gift codes. The first day of the Astrobiology and the Origins of Life conference, hosted by the Biotechnology and Genetics Club of Yıldız Technical University, has reached its conclusion. We would like to thank all our dear speakers who honored us by accepting our invitations, and we would like to thank you, our esteemed participants, as well. You may access the 5th session for your attendance certificate through the link shared shortly. Stay informed about our future events by following us on social media accounts. We eagerly anticipate your presence on the 2nd day of our event. Stay healthy and stay curious.